Got it. Ok, agora já. Ó, já tem pessoa aqui, Alessandra. Ciao Ale.
Come va?
Spero che abbiamo migliorato l'audio, abbiamo fatto delle altre prove. Got it.
Questa mattina era un po' dura.
Sì, pessoa aqui,
Lo so.
Alessandra. Aspetta un momento aqui che Giada, che non sei mai onde c'è. Ciao Ale.
Una cosa strana, aspetta un attimo perché ho un problema con... Voilà.
È fantastica quella, sì, abbiamo capito dopo che c'era dei problemi oggi, ma lei è fantastica questa signora, è 50 anni di ricerca.
Hai visto? Io per quello che ho capito, perché poi la sentivamo purtroppo molto lontana, sì. Ma niente, ha raccontato un sacco di cose interessanti.
Sì, io perché non lo sapevo che...
Ma lei dov'è?
Lei è all'INCA, che è l'istituto di ricerca nazionale del cancro. Sì, ma lei ha fatto 30 anni di UFRJ e 20 anni, e 20 anni di INCA, quindi 50 anni che
lavora sul cancro.
Sì. Alessandra, I would like to introduce you to Cynthia Ferreira. She's our topological data analysis computacional, digamos. We are doing projects on analysis, topological data analysis. She's in San Carlos, USP.
Nice to meet you, Cynthia.
Nice to meet you, yes. Thank you.
Maybe some other time we will tell you about our research we do with our students. It's very interesting what we found with COVID, but she applied also to cancer, to other stuff. So it's very powerful.
Yeah, I would like, yes. If you have a moment, sometime, maybe at the end of August, because right now I'm going to leave and probably you. But the end of August, September, I would really like it.
Yeah. Okay, so we are.
And by the way, do you know? But you mentioned before about your students. But do you know that there are grants between Brazil and France? And there is a Brazilian assistant professor here in the lab. And just yesterday night, I signed for her some, because I have to sign it as a director of the lab, but I signed for her some letter because she applied it in to this ANR and Campus France grant. And from the side of Brazil, there are 300,000 euros for the groups. Yes.
So if you can send us a link, because sometimes there are things that if you're not in the right path, you don't get the information. If you get more, we can apply,
because you mentioned that.
Yeah, because we apply one with Belgium. I was Faperge. Yes, and I know there is one open with Spain, but this with France sometimes are the channels. So if you send us,
yes, I can send it.
Yeah.
So this one is over, but for the future, just to let you know that they do exist.
Yeah, but the problem is where to find information. You see, this is with France.
I think
France is a little bit different because they already do sometime. Also, for example, for exchange student, some schools they have their own. It doesn't follow the Brazilian. It's their own. They come, they do the. We talk to them and so on. So,
camera.
Yes. One second. I have to finish to put things. So we are already on YouTube. So did you put?
Do you want me to share the screen?
I forgot.
1 second, yes. I send you the link, so it's better to put it on the web page because people otherwise they can connect on YouTube.
Web page, okay.
Okay.
Yes, here I am.
Because now there is no need because she will be online to project there. So maybe it's better to project from mine. She will be here.
Yeah, sure.
You see it, huh?
Yes.
The YouTube link.
I send it by email. Email. Can you give me the camera there to see? So test. Is there a plug for the computer? Tem uma tomada para o computador não ficar descarregado? Sim.
Onde está o cabo?
Vou passar. So we're testing, and but that is connected to the projector, no?
Yeah, but
So I give you to the.
I think connect that.
Yes. One second, we're testing. We're testing. But we can do from yours. We can do from this one. It's easier, no? It's just that. Okay. And where is the plug for the computer? 1 second. We are setting up the. It has to be switched on. Sim, to. Okay, 1 second. We are.
Isso aqui é.
Desconecta. Desconecta o áudio lá.
Mas ela precisa de um outro USB.
Isso eu quero.
Tem que botar aí, botar.
Ah, não, isso não é possível.
Não?
Okay,
É porque ele estava aqui.
eu acho que é melhor usar.
Então volta aqui para cá.
Consegue.
Você passa o áudio para cá, caixa.
Volta pra Não tem espaço que funciona.
Pois é, não mexe o time está ganhando. Esse aqui.
Esse aí, então.
A gente ia mudar, mas.
Mas não vai precisar da câmera, porque esse não vai precisar da câmera, entendeu?
Tá bom.
Não.
Esse aqui eu ainda não consigo botar no site. Deixa eu só.
Então, você não precisa da câmera, não precisa de dois, não?
Pois é. Quer vir para cá?
Não.
Porque aí eu preciso mandar as coisas.
Ok, tá bom.
Eu posso ficar de assistente para as perguntas.
Tá bom. Tá, vai lá. Novamente.
Funciona se for no Mac.
Eu não vou poder colocar o net que vai descarregar e nada.
Sólido, meio. Tem cabo.
Não pode?
Não, vai estar puxando.
No break.
Não, mas não pode dar interferência. Eu devo desligar. Eu devo desconectar aqui. Desconecta o microfone.
Eu fico aqui.
Nossa, é que não vai ter espaço para o.
Arranca o meu, então. Arranca o meu. É esse aqui que tem que é. Pronto.
Isso aí.
Está me ouvindo? Espera, como faz isso? Yes. A gente não está ouvindo eles.
Exatamente, isso é um problema.
Espera um segundo, um momento. Vocês estão ouvindo a gente, mas...
A senhora consegue ouvir?
Sim, consigo. Perfeito.
Pronto.
Thank you. Okay, so let's wait like 5 more minutes. Alessandra, just 5 minutes.
No problems.
Okay, we hear everyone and there is no interference. That's great. We improved from this morning. Sí, yes. So just 5 minutes, people are arriving. So in the meantime, I can introduce Alessandra Carbone.
5 minutes.
I will do a better job this morning. This morning I was not aware I was supposed to introduce the speaker. So the great speaker of this morning, this is 50 years of research, and I did a poor job. So first of all, on a personal note, it's a long time I know Alessandra, and I think the first time we met when I invited her for the first biomass conference in 2002 in Evry, where yes, we met, and Alessandra gave a great overview about some research, ongoing research in biomathematics at the time. It was an event that was one of the first year that the group of École Normale, the bio, was founded. So it was a lot of first time. So I'm very happy to have her here in Brazil and around the world, because we have people from Italy, from some other part of, also from Brazil, not only Rio. So let's say that a little bit of Alessandra. So she did her PhD in New York, in mathematics, and then she did also a postdoc in Vienna, and then let's say that she has a background that comes from pure math, but then she was bridging to the biology, and now she's the head of a group that is totally interdisciplinary. And this group was in Paris 6, but now you changed the name. Now it's Paris Sorbonne, and now what's the new name? It's hard to keep up with the changes in France. But so it's Paris Sorbonne, but it's in Jussieu, the campus of Jussieu, I think at least. And now is a laboratory of computational and quantitative biologists. So I really looking forward for your talk, Alessandra. So please, the floor is yours.
Thank you very much for the introduction, Stefanella. So thanks to all for being here. As far as I understand, there are also students. Do you hear me well? Yes?
Yes,
okay.
Yes, perfectly. Now we don't have any.
Great. Okay. So what I would like to do is to give you some idea of some work that has been done around the deep learning today and proteins. And I will talk about protein interactions and the mutations. So why I thought to give this talk in this manner, because what I would like to do is to speak about machine learning and evolution, try to get close to two questions that are sort of important in computational biology today. One is the reconstruction of protein-protein interaction networks. And the second one is the reconstruction of full mutational landscapes of proteins. And here, what I wanted to say is that given a protein, I will be more precise later, given a protein and given a mutation, we would like to estimate the phenotypic effect of the mutation on the protein. We have heard this morning quite a lot about mutations and many mutations. In this particular case, I'm going to talk about one single mutation and its effect, but we would like to estimate this effect in the cell by computational means. And so why these two topics? Because here in the first question, I'm going to use machine learning. In the second one, I'm going to provide and introduce a method that is not using machine learning, contrary to many other approaches that do use machine learning. But as you will see, and what I wanted to say, especially to the student, is that at times machine learning is just so wonderful, like changed completely our life into this problem of reconstruction of protein-protein interaction networks. But at times it might be used, I'm not saying inappropriately, but as alternative to methods that are much more straightforward and can be built if you just use a little bit of biological information, more than what these tools, the machine learning tools, are actually doing. So I would like actually, my main message at the end of the lecture is, so machine learning is fantastic, and you will see why, but just think twice before trying to develop your computational approaches. So let's go. And the first part also, it's about the reconstruction of protein-protein interaction networks. So I would like to construct protein-protein interactions ab initio. This means that I'm going to use just only structures or just sequences and try to construct the networks of interactions out of this information. So in the past, to construct ab initio, so without using experimental knowledge, like we have seen a lot this morning in the type of network reconstructions that have been presented. So here we are using only structures or only sequences. And what I wanted to say is that in the past, we did a lot of work in learning these protein-protein interactions from structures with large-scale docking approaches. And more recently, we learn from sequences only with large-scale deep learning architectures. So let me say a few words about the type of structures that we are going to develop. So they are PPIs, so protein-protein interaction networks. These are graphs. And you see a protein structure in this case that has highlighted with different colors, different binding sites that this same protein might have with a number of partners. So when we think about the interaction of a protein with one other, or eventually many others, typically we need to think about these interactions that is realized on some site of the protein. But in fact, on that given site of the protein surface, you might have proteins that are partners in the cell or non-partners that do bind together with different degrees of affinity. And depending on the way they bind together, they might actually do things together or not. This is what is the difference between being a partner or non-partner in the cell. This doesn't mean so that the protein interacts with just the partners, but might interact with many other proteins. And we shall see, we can see these things in our computations. So somehow the picture is more complicated than just a graph. You want also, in general, to estimate the degree of this affinity. So the picture is maybe of this type, if you wanted to have an image of it. So what we have done for many, many years, because this type of work in my group started in 2006 and 2007, is to approach the question of PPI reconstruction at large scale by using structures and docking. So the idea here, just to make it fast, is that you take one protein structure, you put it at the center of a sphere, a second protein structure, you put it at the surface of the sphere, and then you you test the way in which these two proteins are actually interacting by checking out different type of positions that the two proteins might just take in space. So the protein in the center of the sphere remains there, but you move the protein on the sphere just by in a grid along the sphere. But once you are in a given relative position compared to the center, then you can start moving in that position also the protein on the sphere in different positions. So at the end of the story, you are going to explore about, just to give you an idea, 300,000 different relative positions of the two proteins in the center and around. Okay, so that the whole surface of the protein in the center is going to be scanned, let's say, with respect to the one on the surface. And when you do consider one relative position with respect to the other, of a protein with respect to the other, you want to estimate the energy function that is actually associated to the interaction, potential interaction of the two proteins in a given position. So you do that, let's say, for any pair of proteins in your data set. So suppose that you have a data set, you want to cross each protein with all others. So without using a kind of hypothesis on what might be the partner or not, you wanted to check them all. So given two proteins, you wanted to check these two proteins in many, many different relative positions, and taking any pair in the data set, you wanted to do the same. And once you have this set of proteins, of protein complexes with different conformations, then you wanted to pick up the one with the best binding energy, and maybe a kind of more sophisticated way of discriminating it, and the best one, and put the value of this, let's say, probability of interaction within a full matrix where you have all proteins against themselves. After normalization, you might end up with a matrix now. If I imagine that I know them, okay, if I put proteins against their partners, suppose that here we have a protein and there we have its partner, we would like to have the highest or the darkest value or color within the matrix exactly on the side of the partner. The rest of the proteins, the other ones, are going to be tested with respect to this one, okay? But as you see, they will have different colors, meaning that they will not probably interact, or they will interact in a way that is less interesting. So there was a lot of work done on this, and work that could allow us also to say things about residue interaction and so on. So we did two experiments, one over more than 2,000 human proteins, and the other one over something about 200 proteins against themselves. For the 200 proteins against themselves, we knew about the results. For the 2,000 proteins, we didn't. And so we were actually helped by the World Community Grid IBM that gave us for some years their computer time. And there were about 300,000 people, little less, that helped us around the world to try to answer to these questions, especially on the 2,000 and 300 human proteins that we were testing. So, but here I'm going to speak to you about what we knew, meaning about these 200 proteins that, that we knew were interacting with their specific partners. And so we took in consideration a data set that was a reference in docking, and this is somehow the type of result that we obtained. And so here I put the proteins with their partners. And as you see, as we said before, we would have liked to have a diagonal, which was ideally the only black part on the picture, on the matrix. Here, definitely, we are not in this case, even though you can recognize the diagonal present in the plot. So what I would like to do is to show to you that today with deep learning, we can do much, much, much better. And life really changed. And this is a good example, I think, about what we can do today that we could not do even 5 years ago. So needless to say, we did a lot of work even recently about that. We learned a lot about the protein-protein interactions. So it was not just time to be thrown away, okay? But you will see the type of results today. So let me go fast on what we can do with deep learning and sequences. Now, with deep learning, I'm not going to use the three-dimensional shape of a protein. I'm going to use only the sequence that you can read on the genomes and that codes for this three-dimensional structure. So I'm losing information, but you will see I'm losing it, but with deep learning, I find enough patterns to extract from the sequences to be able to discriminate protein-protein interactions. So why do I want to work with sequences and why it is better? Because with sequences, I can start dealing with also those proteins that are not folded, for which I don't have structures. For proteins in a human, for instance, okay, but that's true. It's true for other organisms as well. We have approximately 40–50% of the human proteins that are disordered or partially disordered. This means that you don't have a three-dimensional shape that you can reconstruct and you can imagine is going to interact in 3D. So this disorder can be complete. Okay, so the sequence doesn't fold in a structure, and or it can be partial. So you might have parts that are a structure and some parts that are not, but it's only, and this is interesting, it's only within the interaction that very often parts of these unfolded proteins get folded, and then in a kind of three-dimensional structure, and then do interact with the partner. So all these situations can be checked when you are going to work with sequences in principle. So let's see how it goes. So now I speak about sequences, but in fact, when we speak about sequences, these sequences are not really what I'm going to use. I'm going to use probabilistic models that are associated to sequences, meaning that a sequence in the human body, okay, is a sequence in humans, but we can find the same sequence within the realm of the organisms that are living today. So we can gather from other species similar sequences that probably are going actually to have the same function, or in any case are very similar in the sequence. I call them homologous because I assume that they have a common ancestor. So when I have gathered all these sequences, I can look at the similarity of all these sequences with the human protein that I'm interested in. And I can find, when I do try to align the best these sequences, I can find positions in the human protein that are actually conserved in all other organisms. And if the protein has these positions that are conserved, the conservation is a good indicator of the fact that these positions are important for the function or the structure of the protein. So I construct probabilistic models where I speak about probabilities because, well, I speak about frequency of occurrence of a given letter, of a given residue at a given position of the human protein, okay? And I can recognize it and I can build up somehow these matrix of frequencies of given amino acid within a given position in the protein. So I will build up matrices where I have the amino acids here, I have the position of the human protein there. The human protein, you might imagine that is this one. And given a position, I'm going to indicate here the frequency of occurrence within the set of homologous sequences in the realm of existing organisms of a given letter in the set of these proteins. So it's a more complex thing than a sequence of what I give to my machine. It is a matrix that somehow is capturing the evolution of these sequences within the world of existing organisms. So this is what it's provided to the machine. And what the machine does is to look at this matrix here with some specific filters. So it looks, you just imagine that the machine has an eye only on some part, okay, of the matrix. So maybe a matrix, a submatrix of 3 by 3. So it goes on this big matrix here, because it can be very large. It can be 1,000 positions, 2,000 positions, but it only sees 3 by 3 submatrices and goes along this big matrix, okay, with this filter, by trying to capture somehow some pattern. And it does it on one protein, on a second protein, on a third protein that I am giving to the machine to train the machine to look at potential patterns that might be found in this type of description. So the machine does that over a number of proteins and a number of pairs of proteins in particular, and it learns whether there are patterns that belong to the two proteins that might be indicators of this interaction. Okay, so here is the machine that I'm going to present to you today. So you have one machine here, a second one there. These two machines are going to analyze one protein and a second one. And at the end of the story, the result of the first analysis and the second analysis will be put together. And finally, the machine is going to say whether with some probability, the two proteins that is going to look at are going to be partners or not. And each module here is actually doing what I suggested to you before, meaning this screening of the subparts of the original matrix. And well, it's more complicated than that because the machines are sort of engineered structures, okay, architectures, but essentially this is the idea, okay? So we do it, and there is one extra thing that I'm going to use, which is extremely important, and this is why I wanted to tell you, because it is a very important thing. So instead of using, as I mentioned before, just a number of organisms that are providing to me sequences that are similar to my human sequence that you can imagine it's here, okay, and then allow me to construct one profile, probabilistic profile representing my human sequence. What I do, it's something more subtle than that. This is actually what, if you heard, but I guess many of you heard what AlphaFold is actually doing. Well, it's doing similar things. So you take your set of sequences before, and instead of constructing one profile, you construct several profiles by putting together sequences that are within your original set, but they are close to each other. So you're going to obtain many profiles that are sort of representing your protein. So not just one profile, but several profiles. And this is an idea that it's extremely important generally for annotation, for functional identification, for functional classification of proteins and so on. And we have tested in many ways with metagenomes, genomes, and so on. And it works also here. And I will show you why. So what do we do with that? Okay, it's the following thing. Instead of working with one single domain, one single profile that is actually somehow representing a protein, and here you have 3 of them, and then we take the machine that just checks 2 profiles, and each pair of these profiles with 3 interactions. What we do is to take n profiles here, n profiles there, n profiles there, and we take all possible interactions, which are 27, okay? So the machine here, since these profiles are different, then has a landscape of motifs eventually that is enriched. And this turns out to be important. So this is our machine I showed you before, and this is somehow the type of improvements that we might have when many profiles are actually used. And you see that on sort of easy sets, like this one, it's on the East, the results just with one sequence or one profile were already very good. But indeed, we can actually improve them a lot. Here we are in a data set of 5,000 proteins, where I take 2,500 proteins to learn the interaction, and then I take essentially the others to test. And within a number of interactions that are at, around 10,000. And here in this second example, I have many more interactions. I have 55,000 interactions. I take the 10% of these interactions are positive interactions. So I know that the 2 proteins are actually partners, and the rest is negative. So we are using that, and as you see, by augmenting the number of models, probabilistic models representing one single sequence, okay, and we do that for all sequences, we end up to have a large improvement of the type of results from 0 profiles or 1 profile, let's say, up to 20 profiles. So, and moreover, what you see is that actually the results are actually extremely interesting. So let me go to show you now what is the result that we obtain now with the same data set that I showed you before, that we checked with the docking and where the diagonal was not actually so clear. Right now, we see a diagonal that it's very, very good, okay? With some proteins that might not be well detected, some interactions that might not be well detected, but overall, don't go into the details, but overall, the type of distinction that we can create is extremely good. And so deep learning made completely life-changing. So I go faster because I have not much time. So what I wanted to also to say, without entering into details, is that there is a way to extract from the machine information on the place where the interaction takes place, meaning that the machine can actually tell me which type of residue is actually important for the interaction. And this can be done. And in this particular case, here you have two proteins with two interaction sites, the pink one and the red one. And here it's a profile that, a curve that we can use. I'm not telling you how we do it because I have no time, but it's a curve that we can extract from by registering the behavior of the machine while it is looking at this pair of proteins that she did never see before, okay? We take this black protein here, okay? And we look at the peaks where the machine, highlights. And we see that these peaks are highlighting the regions in the sequence that contain the residues, and exclusively the residues that fall into the interaction site with the partner. So this can be actually done at large scales, and this type of results can be actually refined. There are many ways of looking at and analyzing these results. We can extract from the machine not only residues that are important for one interaction, but we might end up to have extra residues that are highlighted, but they do not belong to the interactions. And if we go and look more carefully about extra interactions that can be identified, then, like in this specific case, we find out that there are other type of interactions that involve novel residues, and that these residues have been indeed identified by the analysis that the original analysis. So there is a lot to do with these machines, and even about the way in which these machines are actually extracting the information. So I think that it's an extremely interesting field, and I really suggest to the younger generation to go into that. I'm sure that you already thought about that, but go into this because it will make completely life different in the future. So Stefanella, I'm realizing that I'm using a lot of time. So is it fine? We started later. So is it fine if I go with my second subject?
Yeah, of course, of course.
Okay, all right. So let me convince you now that you have to be careful in using machine learning, though, depending on the type of questions that you want to answer. So here I am going to answer a different question. And the question here is, given a protein, we wish to determine the effect of every possible substitution at every position and of the structure. So we wanted to be able to treat single mutations as well as pairs of mutation and triplets of mutations. But here, the method that I'm going to introduce actually lets you do that, but I'm not going to talk about pairs or triplets. I'm going to talk only about single mutations. And we want to answer to these questions to large scale, meaning that we wanted to do it with one mutation, but also with the second residue, a third residues, and so on, by considering them one at a time. So what these amount to? So you just imagine to have your human proteins. I'm speaking about human because people always like to speak about themselves. Okay, so we take a human protein, okay, and we take any amino acid, okay, in the sequence. And we wanted to see this amino acid change. And we wanted to estimate when the amino acid change, what the protein might actually do in the cell, okay? So that's the question, all right? So I'm using only sequences to do it. And I wanted to show you that we can actually answer to these questions. So we are going to estimate what is the effect of this mutation, okay? So let me color it in this manner. But we want to actually look at mutations for all 20 amino acids. So G is not going to be mutated only on A, but is going to be mutated on all possible amino acids. So they are 20. I wanted to reconstruct a column of this type. And I wanted to do it for all possible mutations. So I wanted to reconstruct such a matrix. And each point in the matrix each entry in the matrix is going to tell me how the protein is going to react when I'm going to take one single amino acid and I'm going to change it with respect to this type of amino acids here at the third place. Okay, so that's what the protein, the matrix is actually telling you. So here is the example that I actually treated on the beta-lactamase, that is the structure that I put before, with this little dot that is red. This was not an amino acid. This was a kind of a drug that was there at that given position. And if the protein works well, it's actually cracks the drug, okay? But it needs to work well. And when I start mutating it, then this work might actually not be realized anymore. So I want to estimate that, okay? And here is what we constructed automatically, okay? And here it's what has been actually realized, obtained by deep mutational scanning experiments. This is the matrix that has been realized experimentally, and this is the one that we can estimate. And as you see, the method that I'm going to talk about is actually providing you this, and we checked it against a number, all in fact, experiments of deep mutational scanning that were available at that time. Right now, we retested with the new ones, and we keep going like, with the results that I'm going to show you. So briefly now, I will try to explain why evolution is so important, okay? And so as we did before, we don't have only a sequence for the humans, but we have the same sequence, for instance, in yeast, in the pangolin, I don't know, in some mushrooms of some type or some bacteria, okay? So we recover all possible, as much as we can, sequences that are in our databases that are similar to this one. And we are going to go to see how this G could be mutated in A, and where, and how many times, and in what manner. And if we do it in a kind of fine manner, then we can answer to our question. So that's what I wanted to show you. So there are two hypotheses that we are actually making, and these are the ones. So the first thing that we need to know, and this is where biology comes in, Okay, so someone said it, the first speaker said it this morning many times, we need to learn biology, okay, and he was right. And because the principles are actually very important and they can be fundamental to inspire the computational methodologies, okay? So it was a very good suggestion to the youngest. So the idea here is that the observation is that mutations appearing very rarely in nature. So not only on men, on humans, but on pangolin, on E. coli, and so on bacteria, yeast, blah, blah, okay, are likely deleterious. So if they have not appeared in other organisms, this means...
Quando a pessoa está nesse booster de IgM, essas coisas pegam cruzado pra caramba.
So they are likely to be deleterious also for humans. And another thing is that more conserved a position is, so more we see a letter that is in humans, but it's also in all these other species. So this position on the other extremes need to be very sensitive to mutations, because you can imagine that evolution tries at all means. I mean, these mutations are random, okay? But if something is going to not to be present, or a residue is going always to be there, this means that these are only the selected individuals that the evolution actually decided to get, to accept, okay? So the point is that if you do not see something, or you see it always, okay? So this, the fact that evolution doesn't accept something else, okay? This means that probably is going to be a good weight for deleteriousness, okay? For a negative impact even on human beings. So the second observation is that when a mutation takes place and it is accepted in some sequence, typically it's not accepted alone. It might be accepted because many other mutations took place. And this is another important concept that biology suggests to us. And does exist a concept, the concept of epistasis, that we are going to use. So here, the effect of a given mutation depends on the presence or absence of other mutations. So the state of the art. So how to estimate, to make a model of all this? So there will be an independent model that we are using. We are actually reusing it because other people did it, and takes a position and estimates the conservation of this position within a set of other positions. So here you have the human being, here you have other species. If you find A that is happening, it's very frequent, but some other group contains another position, so that this model is going to take care of telling you that somehow. And you can estimate that with measures that are very known. This is in the case of a Shannon entropy. So we just use something like that. It's frequency. The second model is a pairwise model, and tells you that one position has something to do, changes maybe necessarily with another position. Here, for instance, in this example, you see that when the change takes place here, from these groups of sequences that contain the A, I go to this group of sequences that contain the D, then I have that two other positions that were actually conserved when A was conserved in this group of species change also in that group of species where the D changed. So I speak about coevolution, And there was there are measures in protein evolution that allow you to estimate this type of changes and take care and highlight when a given position is co-evolving with another one in the full set. So there is a lot of work that has been done in these last years about that. By the way, AlphaFold is using this notion for those of you that are interested, okay? So it's exactly this notion. Without this notion, the things that I've done, okay, they couldn't be done, okay? So that's the reason for which they come now and they couldn't come before. So, okay, so here the state of the art is using this independent model and these pairs stuff. But in fact, the last state of the art was to use somehow more of these co-evolution between positions. So instead of considering just pairs of positions, okay, you can consider, or you might consider, contexts that are much larger, but for doing that, okay, you need to introduce in your model millions of parameters. And they constructed what we call deep generative models, and their performance was really very good. Okay, so this, by looking more of the context on of the background of these proteins all together, you can start to have a really very good result on the construction of these matrices that I mentioned before. So I really have a little time, so I go a little fast now. And I wanted to convince you that you can do without these millions of parameters. So let's go back to the two hypotheses that I mentioned. And we take the first, which is the conservation. And there is a little idea that I wanted to give you, and that will give you an idea of the type of notions that we are going to manipulate and use, even if I don't go really into the details. And the sequences, like the human sequence that we have here, and the other ones, have a history. Okay, so this history is represented by trees, okay, typically, that are going to put together these sequences, and closest sequences they belong to subtrees, okay? And when a sequence is very far away, you have to imagine that is going to be also very distant in terms of its evolution from, for instance, the human ones. For instance, let me take the human here, okay, and another sequence there, okay? Here you see that 2 by 2, this letter might be very different. Okay, there are some that remains, some that are different. If you have many letters that change, you might imagine that this one is actually far away. If a sequence is close by, then you have essentially S that I would have left all black with very few red ones, okay, that indicate changes. Okay, so this is what the tree is actually telling you, okay, that closest proteins are actually belonging to subtrees of the tree. So the point is that you can use to estimate the frequency of what's going on in a given position, like we mentioned before with this independent model, you can use the Shannon entropy, all right? But you can do something better that works much better for this type of biological sequences. Again, you don't have to forget that you are working with biological sequences. So you will see it immediately why this is the case. So suppose that I give you 2 positions in my sequences. So you have here a distribution that is absolutely identical to this one. And because of that, you have that the Shannon entropy would be identical, because it's a frequency, okay, counting. So, but if I give you these 2 trees, you can immediately see that here, to arrange the whole letters here, you need 1 mutation. Here, you need 2 mutations within the tree. You see it, okay? So this means that the trees are actually telling us something more about the way in which these sequences evolved in time. And we're going to exploit this. For instance, look at this. You have these 3, you have these 2 positions that are completely conserved, okay? You have the same letter, and they are associated to these large subtrees because all the subtree is actually keeping the same letter. But here you have that 2 subtrees are doing the work. Here you have 2 letters, but you have 3 subtrees. Okay? So here I would like to say that this green one here, it's more conserved than this one because here there are 3 trees and here there are 2. Okay, I needed only 1 mutation. Here I needed 2 mutations there. Okay, at least. So the point is that we can compute out of these 3 how much work evolution had to do, okay, to realize these sequences in nature. In a way, okay? We can estimate this, okay? So you can do it with mathematics, you know. And so let's use this idea. I will be a bit more abstract now. So what the intuition that we want to capture? Suppose here that we have our human sequence here. And we have 2 positions, I and J, in our sequences. And for the position I, we have that the subtree that is actually preserving S, like this one, okay, it's really small. The other sequences have a different letter. And here, instead, the subtree that preserves G, okay, is much bigger. So what I would like to do in my model is somehow to inject the idea that the position J, this one, that is associated to such a big tree, okay, is much more conserved than the position I, okay, and thus is likely to be more sensitive to changes. Because here, changes could happen very, very early somehow, okay? So this is one idea. The second idea is the following. I am here, I am the little man, okay? And I wanted to look at substitution from S into T or S into V. Suppose that the number of organisms that have a T or have a V is the same. I cannot distinguish in the pool of organisms that exist today, okay, the T or the V, okay? Because I found, okay, only 3 organisms that accept the T and 3 organisms that accept the V. But I have that this T is much closer to S than these sequences that accept the V. So what I would like to do here is to say that to accept the T is easier than to accept the V, because you have to do much more work in evolution to accept the V than to accept the T. The T is much closer to the S within a small subtree than the V, okay? So this is the intuition. And with this intuition, I can define 3 parameters. An evolutionary informed conservation, a frequency of occurrence of a mutation in a family, in these sequences of my protein, and minimal evolutionary distances from S into V, for instance, or into A, by moving within the tree, if you want. Okay, versus some million parameters for generative learning approaches that I mentioned before. So 3 parameters are enough to model this problem. So I'm about at the end of my time. I'll be very fast in 3 minutes ago, very, very fast. So just to let you see that this is not a surprise. So we are using the independent models. So this estimation, one single position. And then we are actually making an estimation of this type of epistatic effects, but I cannot explain it because I am too late, okay? And by doing all this, I can actually capture the concepts that, believe me, that I mentioned to you about, and these are the results. But the model is extremely easy, simple compared to the million parameters. Okay, so look at what's happening. So the pink are us, and the blue is Resselman, that is these million parameters business. Okay, so here you clearly see that there are situations in which we are better, there are situations in which they are better, okay? There are on viral proteins, clearly a field, okay, where we are much better than the method that can be applied out of this deep learning. Okay, so we go up and down. Okay, essentially we do the same. And one important thing is the computational time, because, for instance, in the beta-lactamase that I showed you at the beginning of, with the structure, with this red drug, okay? So it was a protein of 263 residues. And here you have that deep sequence, that this huge machine learning approach is 2,000 times slower than our approach. So the full matrix could be, we could compute the full matrix in 1 minute, 5 seconds, okay? And they actually computed with 2,000 times more time. And there are other intermediate approaches that are much simpler than deep sequence that are also very costly, because again, it's machine learning. They really need to learn and to apprehend the sequences, and moreover, they need to have a lot of information to do that. Something that we don't need. So it takes less than 10 minutes, the tool, to treat any protein from the data set. So here is the things if you wanted to try it. And but the important thing that I wanted to mention, as I said before, do not go always with deep learning, because you might find in your data direct features that reveal patterns much more straightforwardly. And it's really better to think a little bit more about biology before trying to do big things. This is my suggestion to the youngest. And just to acknowledge, these are the people that worked with me on the two problems. So Laurent and Hugues worked on the protein-protein interaction networks, and Elodie and Yasaman on GEM. The second part, the reconstruction of the matrix and so on. And Laurent and Yasaman were two PhD students. And I finished with that.
Thank you very much for the wonderful talk. So let's thank Alessandra. Your last remark, I found of your last remark, one thing is just using data. The other thing is theoretical modeling. You know, the two things are complementary, but not just data analysis without a theoretical knowledge of the phenomena of understanding biology. It's very important, yes. So there was some question. In the chat, one was, can you, one second, when you have one profile you are using the probabilistic idea you showed before, but how do you construct more than one profile?
Yes, so you can do it. It's very simple. So first thing you can imagine is to take as many sequences as you can that are in your space of homologous sequences, okay? Just you go down with the sequence identity. So you gather all these sequences. And so what you do is instead of using them all together to construct your profile, so you take groups of sequences. Let me see, just to be more very practical. Okay, just imagine that you have your sequences and that your sequences are organized with the tree. So you could imagine to construct a profile just for this subgroup, okay, of sequences. You take them and you construct a profile. Then you take these ones and you construct a second profile. And then this one and you construct a third profile. So the point is that when you look at your space of sequences, I mean, here I put a tree, but I don't like to put a tree on the space of sequences. But I like more to think of them as points in a multidimensional space. So when you look at this multidimensional space of sequences, you see that your sequences are sparse in this multidimensional space, but some of them are closer together. So you put them, you take the ones that are closest and you construct a model for them. And then you go to another one that are closest and you construct a model for them. And by doing that, what you are doing actually is to extract conserved parts of these groups, okay, that are separated, that are different. And these conserved parts are actually determinants of function and structure. So sampling your space of sequences, it's extremely important, okay, in the questions that you wanted to answer, to extract the best of the biological information that is inside sequences. I hope that I was clear.
Yes, he say Anderson, thank you. Okay. Okay, so we have a little bit of to rush. Let me think that it's hard to juggle between YouTube and Zoom, and so maybe there are some other questions on YouTube that I'm not seeing right now. Eventually I will send you because the computer froze on the Zoom and I can't see the YouTube. But thank you very much. It was great. So would it be possible to have the slides to put on the website?
Yes.
Possible? There will be a lot of very interesting materials. So I'm sure that people will be able to use it more.
Okay.
So
thank you very much.
Thank you very much. So any question from here? So no, we got to, okay. So no, we are in a rush. So thank you very much. And so with we are going to the next speaker. That is not me that introduce. I am not going to take the privilege to introduce everyone. So yes, yes. So it's going to be Francisco Lopez to introduce the next speaker. 1 second.
Tá bom, é que eu cliquei. Mas ele já está. Great. Good afternoon. My pleasure to introduce Dr. Jean-Marc Schwartz. He has a degree in engineering from the École Centrale Paris. He was a research associate at Max Planck Institute for Biochemistry in Germany. He received his PhD in computer science from Laval University, Canada. He was a postdoctoral research at the Kyoto University Bioinformatics Center, and he was Physical Life Science Interface Independent Research Fellow, Faculty of Life Science, University of Manchester, and now he is Senior Lecturer, Faculty of Biology, Medicine and Health, University of Manchester. Professor Jean-Marc, thank you very much. You have the words. Thank you.
Thank you. Thank you for the nice introduction. I hope, can you all hear me clearly, or do I need to go closer? Is this okay?
Sorry?
Suppose yes, you can hear me clearly. Okay.
Perfect.
So let me share my my slides. Just a second. Okay, I suppose it works. Okay, so hello everyone, and thank you again for the invitation to present. Sorry that I cannot be in Rio, but I hope it still works well like this. So I wanted to present some of the work we did in recent years, and the topic I chose was to focus it on what I call systems and network approaches for the investigation of human diseases. So the idea is that, so the main idea I think of the presentation is that in recent years we know that we have more and more technical capabilities to produce data, different types of omics data, including genomics, transcriptomics, proteomics, and so on. But the main challenge is often to be able to exploit this data and in a way to make it usable for biologists and medical and clinicians so that they can use it towards advancing some treatments, for example, finding new drug targets, biomarkers, or patient stratification, which means, for example, being able to personalize treatments for different types of patients. So the challenge is often, how do we make all these data available or understandable in a way that biologists and clinicians can use the data? So that's essentially the main idea behind the presentation. So I've chosen to cover several examples to give you an idea of the kind of tools we can use for this goal. And the first example was a project that we did on systems biology of skeletal diseases. So this was a large European project where we had several partner labs from different countries, and they were producing, they were analyzing samples from patients affected by many types of bone or cartilage diseases, including some very common diseases like arthritis, but also more unusual diseases like diseases that affect usually a single gene, monogenic diseases, but that can affect, can have many effects on bone development or growth, for example. So when we get this kind of data, the first thing that most people do is to first compare gene expression. So for example, we have samples in healthy versus diseased, like here arthritis samples, and we start by looking at which are the most over or under expressed genes in the disease compared to the healthy sample. So this gives us an idea of individual genes that could be involved in the disease, but of course, what we can learn from it is somewhat limited. So how can we go further? Well, the next step is usually to use a gene ontology. So I suppose many of you are familiar with the gene ontology. So it's an annotation that is defined and associated to all the genes that gives us some idea of, for example, the cellular processes or localization or molecular function of different genes. So we can look for enrichment in gene ontology annotations instead of just the names of the genes, which gives us already a little idea about the functions that may be involved in the disease. But if you've used gene ontology, I'm sure you've probably encountered this kind of results. You know that you often get very long lists of functions, and they are often not very informative in the sense that you have very general functions like anatomical structure development, but probably we already knew that because it's a bone disease. So often what we can learn from this type of analysis remains relatively limited. So there's one way we can improve the, let's say, information of gene ontology analysis. It's to use redundancy reduction. So there's a nice tool here which I'm presenting. So it's not by us, but I'm presenting it because it's quite useful when you analyze Go terms. It's called Revigo. And what it does is essentially it reduces a long list of Go terms to a more representative subset of the terms based on semantic similarity measures. So instead of having a long list, you will have a shorter list which contains non-redundant terms. So that already helps in the interpretation. But then, in many cases, it still remains a bit insufficient. So the next step of what we usually do, and many people do, is to look at pathways. Because unlike Go terms, biological pathways are experimentally validated sets or maps of proteins which contribute to a specific function. So in a way, pathways are often more informative than just Go terms, because they give us the full context of the function. Essentially, we can see all the genes and all the proteins involved in a particular function. However, when we start, so there are many databases, and maybe you know some of them. There are many pathway databases like KEGG, Reactome, or Wiki Pathways. But when we use them, we often get into the same type of problems that we get with the gene ontology. The problems being that there are many overlaps between pathways, there are duplications, and also there's the problem of size. So many, sometimes you get a pathway that contains like 200 genes, and you get another one that contains 2 genes. So they're not really comparable, and again, quite difficult to interpret. So in order to let's say improve or resolve this problem, we've developed an algorithm which is called a set cover algorithm, which attempts to reduce the redundancy found in results of pathway enrichment analysis in a way that, well, you remove the redundancy, so in a way you avoid having overlaps between pathway. You control the pathway size, so you try to have a homogeneous set of pathways which are all of more or less the same size, and at the same time still maintain coverage. So we don't want to lose genes in the process. So just to give you an idea, without going in the details of the algorithm, what the algorithm does is essentially, if you look at panel A, so this is essentially the situation that we typically have in a pathway database. You see, so there are large pathways like the red one, there are small ones like the orange, and there are many overlaps. For example, the green one is completely included in the red one. And what we want is to get a situation like D here, the panel D, where you see that you no longer have overlaps between pathways, you have pathways that are more homogeneous, have more or less the same size, and still we minimize the loss. So here we lost one gene, but we don't want to minimize to lose too many. So that's essentially what our algorithm does, is to reduce the set of pathways to a more consistent set that is then more suitable for enrichment analysis. And here is then again an example applied to arthritis, where you can see that you get more specific. So the idea is to get more specific terms, and also terms that will be more understandable by biologists. And still we get the full list of, for example, genes or proteins included in these pathways, and the enrichment with the p-value associated. So it means that here, all these reduced pathways are enriched in arthritis in this case, and we can then see more specifically what these pathways may be doing. So that was pathways. So that's still not enough for many people. So we wanted to go one step further, and the next step is to look at now networks or subnetworks. So here I'm referring to work that was published by Barabasi, and I guess if you're a bit familiar with the network literature, you probably know his name. He's probably one of the most famous researchers in the field. And essentially what Barabasi showed is that in many diseases, essentially in most diseases, we see that proteins involved in the same disease have an increased tendency to interact with each other. So if we connect all the proteins according to the interactions, and then we look at, for example, specific proteins overexpressed in a certain disease, we often see that they build modules. So they tend to be connected. They are not just randomly distributed over the network, but they tend to be connected, and therefore to build what we call a module. So we can use this property again to try and better understand the mechanisms that may be involved in a certain disease, but we can also use it in order to predict new disease-associated proteins. So for example, if you look at the panel here on the left, D, if we know that all the proteins in blue are related to a certain disease, then it's quite likely that the white one here will also be related, because it's very strongly connected to known disease proteins. So again, that's something we can use in order to get more specific information about networks involved in some disorders. And so what we've done is that we've tried to slightly improve, again, the previously known algorithms by adding something called a Phenome Network. Because one of the issues that we found, so if you're using general protein interaction networks like the ones shown here, the edges, so the connections between the proteins, they come from many different types of experiments and many different contexts. So you may have interactions that have actually nothing to do with the disease you're looking at. Just to give an example, if you're looking, for example, at bone diseases, you may have interactions that were detected in the brain. And then, of course, they are not really relevant to cartilage, for example. So to try and avoid this kind of problems, what we've done is to connect the protein interaction networks. You have the protein interaction network shown in green here. We connect it to a phenome network, which is then a list of phenotypes that are specifically relevant to the disease that we investigate. So essentially, each protein is connected to the phenotype in which it's involved. And then our algorithm, which we called Phenome Expressed, involves some kind of random walk. So essentially it means you start randomly from a node in the network, and then you kind of go through your network randomly. And after a certain amount of time, you end up somewhere, and you just record the probability of reaching a particular node. But because the protein network here is connected to a phenome network, it means that we will more often reach nodes that are also connected by phenome. So in a way, it's a way of improving the module detection by favoring nodes that are related to the same phenotypes. That's the main idea. So if anybody is interested, so this algorithm is now available as Cytoscape application. So Cytoscape is one of the main software that is used for network analysis. So if you are familiar with Cytoscape, you can look at, you can search for Phenomescape in the list of applications and then install it. And then the result is you get something like this. So essentially, what you get is a list of, or a network, a subnetwork of proteins that are differentially expressed in your experiment. So for example, the red ones are overexpressed, the green ones are underexpressed, and at the same time connected to the phenotypes that you're interested in. So in this case, decreased or increased susceptibility to arthritis. So, so just to continue a little bit on how we applied this, all these tools then to arthritis. So, well, maybe I won't insist too much on what arthritis is. You know, it's a common disease involving cartilage degradation. One of the important things is that there is no real treatment to stop or prevent arthritis. We can only treat the symptoms, but not the disease itself, which is why it's still an important subject for research. So when we applied Phenomescape to osteoarthritis data, we found a few new interesting subnetworks, which are then dysregulated in damaged osteoarthritic cartilage. Here, for example, this is related to the extracellular matrix organization, and we can see a certain number of proteins which are all highly connected and all dysregulated in arthritis. Another module here, which was related to blood, but includes also a certain number of well-known proteins like serpin. So perhaps more significantly, we then applied these algorithms to a selection of 44, in this case, patients affected by osteoarthritis, and also control group of 16. And we found that, well, the first thing we found is that, well, luckily, the PCA shows that the non-OA group is well distinct from the OA group. But crucially, we also found that among the osteoarthritis patients, by combining the network analysis with other types of clustering analysis, we found that there were two subgroups which are quite clearly distinct, which you can see here. Well, it's maybe a bit small, but on the B panel, you can see that there are two subgroups which are quite clearly separated. So what it means essentially is that, so using the network analysis, we can see that these two subgroups, which are both osteoarthritis, are quite different, but they are not different from other types of factors. So if we look at other types of clinical data like age, body mass index, and so on, there's no clear distinction between group A and B. So the distinction only occurs if you look at some specific genes, and here we can see, so we identified 10 genes in particular, where the two subgroups are quite different. So you can see again on the panels all the 10 genes, and group A is in white, group B is in gray. So all these 10 genes are clearly enable us to clearly separate the two subgroups. Also, we find that, so essentially, if you look at differentially expressed genes between subgroups, you find that you have more differentially expressed genes than if you treat osteoarthritis as a single group. So for example, you have 1,800 genes which are common to both. But if you treat everything together as a single group, you only add 44, whereas if you treat them separately, you see that you add lots of other differentially expressed genes. And here are the specific subnetworks that are distinct between the two subgroups. One includes, for example, complement activation. Another one involves TLR4 signaling, and another one involves Wnt signaling. So maybe this one is actually quite clear to see, that these 4 proteins are all very highly expressed in group B, but not in group A. So before I move on, so mainly, essentially, I would say the main message between that is for this section is that, so essentially it's the network analysis that enables us to distinguish 2 subgroups of osteoarthritis patients, which could not be distinguished by other methods. And also the network analysis enables us to show what are the specific pathways, or let's say modules, which are different between the 2 subgroups, which is why I would say my main message here would be that we think that the network analysis, well, this shows that the network analysis adds some understanding which cannot be found using more classical methods. And just one more slide here on this particular topic is that we've then also taken all the samples that we got from all the different bone diseases and put them on this graph, which is called TSNE. Again, I don't insist too much on how it works, but essentially it will put more closely the samples which are more related. And you can see that, again, this enables us to quite well distinguish all the different diseases. Like here you have osteoarthritis in which is called OA. I'm not sure if you actually see my mouse, so otherwise I have to tell you where I'm showing. You have rheumatoid arthritis, which is called RA here. And maybe also the nice thing with this tool is that now if, for example, a clinician comes up with a new patient or new sample and they don't really know what it is, what we can do is simply put it on this map, and then we can tell essentially, well, this sample is more related to, for example, this group of known disease or this other group. So it can be also a useful tool to identify new patients. And and just to show that, so we've put these results and these tools on a website which is called Skeletal Viz, which you can find at the address shown below, phenome.manchester.ac.uk. So you can, if you're interested, you can try it yourself and then visualize these results. So all these different types of results, including good terms, pathways, subnetworks, for all the different diseases that we analyzed. But of course, it only covers bone diseases, so not things like cancer or other types of diseases. So the conclusion for this first part is that so what we've shown is that genome scale expression analysis leads to more global description of dysregulated processes in diseases. But perhaps the most important message would be that we think that if you add network analysis to other types of omics data analysis, it enables you to give a more specific identification of the functions or the processes which are involved in a particular disorder. And crucially, we showed, at least in the case of OA, that it enables better patient stratification, because we saw that here we could distinguish between different types, at least 2 types of arthritis. So maybe the next step would be to see if there's not a way we can also personalize the treatment to these 2 different subgroups. So maybe group A would benefit from a different type of treatment than group B. And that's then, of course, the ultimate goal of this type of work. Okay, so that was the first part I wanted to show. So I'm just checking if there's no immediate questions, so then I will just go on. So the next part I wanted to present is then, so here we were mainly focused on gene expression. Another type of omics analysis is then proteomics, which is also more and more widely used. And so I wanted to show also how we can analyze proteomics data and learn more from different types of proteomics data. So again, going back to networks, we know that proteins inside the cells form interaction networks, and we very often represent them like this. But there's one new element when you're using proteomics, is that you can also use tools called Phosphoproteomics, so which we're using now in Manchester, because it helps us to understand the dynamics of the network. So essentially, if you have a protein network, what usually happens in the cell is that there will be some kind of ligand or signaling pathways where a hormone is when a hormone is applied or drug treatment is applied, which then connects to the cell, and this will lead to some kind of signaling cascade inside the cell where different proteins will be phosphorylated. And actually being able to measure these phosphorylation is very useful because it gives us an information on actually what happens dynamically in the cell. So unlike if I go back one slide, I would say this is a static network. We know that all these proteins interact, but we don't really know why, when, or what happens. Here, we know a bit better over time what happens when a particular ligand binds. We can see that different phosphorylations appear on different proteins. So this helps us to understand the dynamics of the pathway. However, again, due to a lack of phosphorylation-specific methods, very often when people analyze proteomics data, they just analyze the proteins. And this is not really enough to understand what happens dynamically in a pathway. Because if you look, well, again, we can go back to gene ontology. Let's say you have a protein X and you look at what is its function in the gene ontology database. You get things like this. You know that it's involved in a particular process, it's involved in a particular pathway or in a complex, and so on. But it doesn't mean that this protein will always do all these things it's involved in. So here's just one specific example is if we look at the RAF1 protein, when this protein is phosphorylated at a particular site here called S338, then we know that it's involved in inhibition of cell death or promotion of cell growth. But if it's phosphorylated at another site like S29, then it's actually inactive. It's not doing anything. And if it's phosphorylated in both sites, then we actually don't know what it's doing. So you see that depending on the phosphorylation state, the protein can do very different things. But if you ignore the phosphorylation state, you will just take the whole thing all the time, which again may not be really useful now in terms of understanding what is happening in the system. So that's why we tried to develop a method that helps us to interpret phosphoproteomics data more specifically rather than just proteomics data. And essentially the method is based on the same principle as the previous method, so as the Phenome expressed, except that here we extended it by adding a third layer of data. So in the if you remember in the Phenome Express algorithm, we had two layers of data. One was the protein or gene interaction network. The other was a phenotype network. So here we're using now 3 layers. So first layer is still the protein interaction network, shown in red here. We add a second layer of function annotations, which is, we could say, more or less like the Phenome Network. But then we add a third layer, shown in blue here, which is a layer of phosphorylation. So this is coming from the experimental data. So what we do is that we build clusters based on the profile of the phosphorylation. So the assumption would be that proteins which have the same dynamic profile over time are more likely to be involved again in the same process. So we build this third layer here of phosphorylated sites. And then, well, more or less the algorithm works in the same way as before. We have some kind of random walk, which starts from a specific cluster and then walks through the network. And then we measure how often do we reach a particular node. And the idea would be that if a node is reached more often, it's more likely to be related to the source of the random walk. So to illustrate what this gives concretely, so for example, if we apply it to, so this is just applied to a small validation data set based on the MAP kinase pathway, because we know what it's doing. So this is the network. So essentially what we do, we build different clusters based on the dynamic profiles of the proteins. You can see, for example, cluster 1 and 2 are the ones which we find interesting here. They are all unphosphorylated at the beginning, and then they become phosphorylated later. They switch from red to blue. Well, according to our analysis, we can then see that these 2 clusters, 1 and 2, are actually correctly attributed to negative regulation of response to stimulus, which is what we're doing here. We are stimulating the pathway and we get actually a negative feedback loop, which switches the pathway off. So this is promising. It looks even more promising if we compare it to standard protein enrichment analysis. So on the right here, you can see the result of the standard analysis. And you can see that, well, first, it's much less specific. We can't see many specific annotations to specific clusters. But furthermore, it's actually wrong because it tells us a positive regulation when in fact we have a negative regulation, whereas the random walk algorithm gives us correctly the negative regulation. So another way we then tried to further validate this new tool was to compare it to a database called Phosphocyte Plus, which is a manually curated database of more or less 4,000 phosphorylated protein annotations. And essentially, we see that in most cases, our results tend to match what Phosphocyte Plus gives, with perhaps a small exception here. Cell differentiation is a bit under detected, but so let's say in some cases we can still under detect. Then there may still be things we don't detect properly, but at least we don't detect false things, which is, I think, perhaps more important. And then we further applied this tool then to a new experimental data set, which was generated by one of our collaborators here, which involves 2 types of ligands activating EGFR signaling. So on the right here, you can see all the different phosphoproteomics profiles that we observe. So we distinguish 6 different clusters. And then we apply, again, we apply our tool to these 6 different profiles. And then you can see on these graphs for each cluster, what are the terms which are enriched in these different clusters. Importantly, both give us regulation of ERK1 and ERK2, which is what we know happens. So this is also correct because it's what was done in the experiment. And again, important thing is that we can see that both clusters associated to this ERK1 and ERK2 regulation give us a number of other relevant terms like establishment of protein localization, positive regulation of MAP kinase, endosomal transport, and so on. And again, just to be systematic, we systematically compared our results to the standard protein annotation analysis, which we see gives us either very general terms, which are not really specific to our experiment, or in this case, actually sometimes even wrong terms. So I would say in all cases, we see that applying this kind of tool to specific phosphorylation sites rather than just whole proteins gives a much better analysis or much better understanding of what is happening in the experiment. So again, a conclusion of this part would be that if you want to analyze if you want to analyze proteomics data, it's much more useful to look at a site-centered approach, so to actually take into account how these proteins are phosphorylated at specific time points in the experiment. This gives much more specific or exact annotations than just looking at the entire gene or protein. And in a way, our method is the refinement of the most commonly used enrichment methods and gives us more information about site-specific function. So again, it should be a gain towards better understanding what happens in different signaling pathways. So in this case, I would say it's mostly relevant to signaling pathways. And again, making a short break. So this was the second part. So we've looked at gene expression analysis first, proteomics, phosphoproteomics data second. And so I think I have about 15 minutes left. So for the final part, so since I know that some people maybe in your team or in the audience are also interested in viruses, so I wanted to add some presentation of more, let's say, I would say, so this was more like a bit of a side project in the last 2 years. But as we all know, because of the COVID pandemic, perturbed many of our lives and of our work. So we decided to see if we could also apply network tools and analysis to this kind of viral epidemic network. So that's a slightly different subject, but I hope you're still interested in it. We know that many types of computational models were used during the pandemic, COVID pandemic, because they help to predict how infectious diseases can spread in population, and also to understand what will be the effect of different mitigation tools or measures. I would say more importantly is perhaps not the prediction that that these models give, because in many cases people say actually the models don't really predict what happens, but it's more about being able to understand what happens. Now, if you have a model of an epidemic disease, it helps you to understand what is the influence of different parameters. So what happens if, for example, the virulence of the virus changes, or if we take some social, reduce social contacts and so on. So it's more the idea that if we have epidemic models, it helps us to investigate different scenarios and then take the appropriate measures. So of course, if the model prediction does not happen, it can be a good thing, essentially, because we use the model to try to prevent, you know, the worst case scenario to happen. So among the models that were used in this pandemic and other types of epidemic diseases, I would say there are two main types. Maybe the most commonly used models are what we can call continuous models, which are based on differential equations and which assume essentially that, well, the population is quite homogeneous and that people kind of meet at random. But this is often not very realistic. So another type of models that can be used is, again, network-based models. And you've understood that networks are one of the main themes of the whole presentation. So of course, we're still using network also in this project. So the reason why we believe that network-based models are more accurate than differential equation-based models is that it's quite well known, again, thanks to the work of Barabashi, that social contacts are not random and not homogeneous in a population. In fact, we know that, you know, most kind of social network science tell us that there's a relatively small number of individuals which are responsible for a very large number of interactions. So these people can be then called hubs. And on the other side, the majority of individuals have a relatively small number of interactions. So again, if you compare a random network on the left to a real network, the real social network looks more like the panel on the right, which is called a scale-free network. And you have a few hubs. You can see the blue nodes or gray nodes here are hubs which have many connections, but the majority of nodes in red have actually quite few connections. So this is a much more accurate representation of real social contacts. One of the problems with ignoring this kind of properties is that if you ignore this particular structure, then you cannot test mitigation strategies which take advantage of these features. And this is perhaps one of the problems in how we responded to the pandemic. So to try and see how these network effects can affect the response, so we've built relatively standard infection model, which is called SIR model. So essentially it means we have three types of states for the nodes in the network. So initially the nodes are called susceptible, which are the blue ones, which means they are healthy, but they can catch the disease. Then we start by infecting a single node in the network. So assuming, for example, a traveler arrives and introduces the virus in the population. So a single node is infected initially. And then susceptible nodes can be infected by the neighbors according to a certain probability distribution over time. Infected nodes recover after a given number of days, which we took about 14 days, or 14 days in this case. And then after infection, the nodes switch to R state. The green nodes are recovered nodes. And we assume that these nodes are immune to reinfection for a certain number of days. So again, not forever, because we know now that people can catch COVID more than once. So after a certain number of days, which we call immunity length, these R nodes can become susceptible again. So I would say first interesting result that we found is shown here on this slide is that we can get different types, at least two main types of outcomes. So so I should clarify that again. So because this is not a deterministic model, it's a stochastic model because it's based on probabilities, which means that every time you run this type of model, you get a different result. And what we can see is that now starting in the same state, you can end up in at least two very different types of end state. One of them is a state where there has been no propagation at all. So essentially in about 25% of the simulations, you end up with finding just maybe 1 or 2 nodes. So this may be the initial infected node, just infected one other, and then it stopped because they didn't have time to infect other people. In other types of simulation, we find that actually most of the nodes have been infected, and there just remain a few people who were not infected. So this is what we call herd immunity, which means after a while, well, most of the people were infected, and essentially, well, some people remain susceptible, but they are protected because they are connected to immune people. So one of the first questions would be, so what, so why, you know, why does this happen? Why does, what makes the switch between the state in A to a state in B? So we decided to look at the role of hubs. And one of the first things we did was to look at what happens after a hub is infected. So this you can see, this is shown by the blue and purple distributions on the right. So after a hub has been infected, a peak happens very quickly. So in the next 5 to 10 days, you get a peak, which shows that hubs actually play an essential role in, you know, increasing the spread of the disease through the network. If you compare the blue distribution to the red one, which just shows, you know, what happens in all the simulation, you can see that there's a clear shift. So based on this, the next question will be, what happens if we try to reduce contacts in such a network by primarily reducing contacts from hubs? So which is why we compared 2 mitigation strategies. So one is called random mitigation, which means we just reduce contacts randomly, which means we just remove edges randomly. The other one is called hub mitigation, where we only remove edges from hubs, but we remove the same number of edges. So this is important. In both scenarios, the same number of edges is removed, but here it's only removed from hubs, whereas in random case, it's removed from everyone. And you can see that with the hub mitigation strategy, there's a clear difference in that the peaks happen much later than in the random mitigation. So the random mitigation is shown here in brown, and the hub mitigation is shown in orange. So you can see there's a clear shift. The peak comes much later. And not only does it come later, but it's also a much lower peak. So if you see here the maximum, so this graph shows the maximum percentage of nodes infected at peak. So this is on the left is without mitigation. Hub mitigation is about 20%, random mitigation is about 30%. And you can see here more or less what it looks like in actual simulation. So in the hub mitigation, you can see that you get lower peaks, and also the peaks are delayed. They come later and they are lower, which is essentially what we want. Now, what most people were saying during the pandemic is that we want to flatten the curve. So avoid healthcare being overwhelmed means we want to reduce the level of the top of the peak and spread it more over time. And we can see that this is much better achieved by hub mitigation than by random mitigation. Just checking the time, yeah. So these graphs shown here, they were based on a single peak, but then of course we can repeat this kind of analysis over multiple peaks. So if we add a finite immunity length, which means that people who were already infected can become reinfected after a certain number of days, then we get curves like this, which show that there can be multiple peaks, as we've observed in the real world. So of course this becomes more, I would say, more complicated to analyze, but we can still do interesting things with this type of data. For example, here we've then also looked at how could this affect vaccination strategies. And again, we've tested two different types of scenarios. On the top panel, what we could call random vaccinations, which means we just vaccine that are distributed to everyone randomly. And at the bottom, what happens if we administer vaccines in priority to hubs? So again, this may be a bit difficult to see on this screen, but you can see, at least you can see that on the top panel, the red curves, which are the infected people, are still visible even after 2 years. Here you can still see some red, whereas on the bottom panel, the red curve has essentially almost disappeared. So you can see that there's a much lower number of infected people in the hub vaccination than in the random vaccination scenario. We can see it a bit better on these graphs. So again, this compares random vaccination is at the top, hub first vaccination is at the bottom, and then we compare different types of metrics in the result. Perhaps the most interesting one is that, so the hub vaccination is the only one which can give us results with 0 infected people. In the random vaccination, we never get a zero result. Now you can see that, so this panel B here is the percentage of infected people after 2 years. You can see that it still goes up to maybe 4%, but in the hub first vaccination, it's almost 0 in all cases. So that's already a nice result. Of course, I should maybe add one limitation is that in this study, we assume that the vaccines essentially are perfect, so that they provide perfect protection. Of course, we know that this is also not the case. So the vaccine effect can also diminish over time. So this is still, let's say, a perfect case, but still, I would say the main point is again to show that there's a very strong difference between hub first and random vaccination. And we can see also here in the third panels, so this is the maximum number of reinfections. You can see that in random case, it usually goes from 6, it can go up to 6 or 8. In the hub first case, it goes to maximum of 5. And also there are shorter peaks. So this is the percentage of days for which more than 1% of the population is infected, which can more or less say there's a peak. Again, in the hub first case, we get peaks of up to 40 to 50 days. In the other case, you get 60 to 90 days. So in essentially all these 3 metrics, there's a clear difference between the two. The only metric which shows no difference is the one on the left, which is the maximum percentage of infected at one time. Here, there's no clear difference between the two scenarios. So again, take into account hubs looks like a good idea for vaccination. And just to finish, so this is my last slide, but this is still, let's say, very preliminary. We more recently, we also asked whether we could use this type of model to try and predict what happens when you have more than one variant in this kind of system. And so I'm just showing two results here on the left. So here now you have, let's say, 2 viruses instead of 1. So you have variant 1, which is in red, variant 2, which is in orange. So if you take, let's say, very close infection rates, now here you have R2 versus R1 is just 1.1, which means the second variant is just 10% more infectious than the first variant, then you can see that in this kind of system, both variants can quite easily, well, cohabitate in a way, that you have peaks of both variants even after 6 months. If you take, let's say, a higher difference between the two variants, let's say here R2 is 50% more infectious than R1, you get fewer orange peaks. You get more red peaks after a while, but you can still get a few orange peaks. So in a way, this is also interesting, but maybe too early to conclude. So in a way, it shows that even, let's say, a less infectious variant can still continue to propagate in this kind of system. So it would be interesting then, the next question would be interesting again to see whether this is due to the role of hubs. So does it mean that perhaps if the less infectious variant reaches a hub, it will again propagate more? So in a way, it remains to be determined whether hub effect can override differences in transmissibility. So this is still under investigation. But the other points I would say are quite clear, is that now we show that network-based epidemic models are able to simulate a wider range of real-world scenarios, and also to test more types of mitigation strategies than, let's say, models based on homogeneous compartments. We can see quite clearly that under the same conditions, different types of outcomes are possible. So this is why it's perhaps a better tool to see all the possible scenarios that can happen. And also that in all cases, it appears that prioritizing hubs or mitigation or vaccination appears to be more efficient than just random strategies. Of course, this is, again, let's say, a scientific result, but then in reality, if we wanted to implement this kind of strategy in practice, we run into lots of other types of problems, mainly how do you decide who is a hub and who is not a hub in society? This is, of course, not easy to implement in practice. And yeah, so these are just the two publications that covered these results, except the variant work, which is not published and not finished yet. And some thanks to the people who contributed to all these these projects. Joe Watson did the Phosphoproteomics analysis. Jamie did the bone analysis, who was responsible for the pathway enrichment analysis, Helena for the COVID, and collaborators from Manchester, and also, of course, thanks to our collaborators in Brazil, Mauricio and Marcelo. And that's the end. And I think maybe short time for questions.
Great. Thank you for your very, very good talk. We learned a lot. Let's see if we have questions. Questions here. Professor Fabrício has one question.
Hi, Jean-Marc. This is Fabrício. Thank you very much for your great presentation. Thank you a lot. I have a question. Is it correct to say that about phosphorylation, right? Is it correct to say that phosphorylation can change the structure of the underlying PPI network, and because of that, change the dynamics of the system? Is that correct?
So I wouldn't say, no, I wouldn't say that it changes the structure of the network. So the network remains the same. The question is now which of the connections in the network are actually active at a particular time. So that's what the phosphorylation enables us to say. So the network is still the same. So we're not adding any or removing any connections in the network. But the phosphorylation enables us to say, no, at, let's say, time point 0, these two proteins are actually doing something together, because we see a change of phosphorylation that happens here and then here. And at the next time step, maybe it's these two proteins. So that's what the phosphorylation data adds now, is understanding of what kind of dynamics happens in the network over time, instead of having just a static network. But still the network remains the same. So the network itself is not modified. Hope that was what we asked, but I don't understand so clearly the questions from the audience. Sorry.
Thank you.
Simple question. So the definition of network, because for us, a network, there is a connection because they are connected. So when you can ultimately define a network just when there is when one phosphorylates the other. So if you just consider this network, the network of active phosphorylation, can you, this is dynamics, like you say, this is not the same, this is evolving. So do you see how this structure, this network of the one that is phosphorylated might have? Yes. Sorry, disappeared. This structure, the phosphorylation network, the one that at that time they're connected, does this has an effect? I think it was one of the levels that you showed, if I am not mistaken. You showed the third level, the first and the third level.
So I'm not sure if I understood everything. From the sound is a bit low. So yes, I think you were asking if we could observe changes.
It's similar question that he was asking, actually. It's very similar. So you could, because I think what you describe as a network is the possibility of a connection between two.
Mm-hmm. Yeah.
And now let's consider when there is a connection. So one is acting with the other.
Mm-hmm.
So if you consider this dynamic network, so if you consider when there are some network, when there is acting the phosphorylation, in some configuration, let's say, there is something happen and some other doesn't happen. If you consider the frozen part, let's say, of so it's similar to this question, actually.
So I think the answer is, so we did not try to visualize directly, which edges are active or not at a particular time points. This happens more through so we do that, I would say, more indirectly through the clustering. Because what we do is that we build clusters. So we take all because in this data you have usually hundreds or thousands of proteins. So we build clusters where all the proteins that follow the similar trend will be in the same cluster. And then I would say indirectly, the idea would be that, or at least the hypothesis, so it's still a hypothesis, but we think it's a good hypothesis, would be that if proteins follow the same dynamics, probably it means they interact somehow, because otherwise, if they were completely disconnected, it would be very unlikely that by chance they follow exactly the same pattern. So I would say it's indirectly that, we could say that proteins in the same cluster follow the same pattern over time. Probably they interact then.
So the phosphorylation.
Hope that answers maybe partly your question.
You could also say that sometimes the phosphorylation change with your network. Is it also possible this underlying network that could be changed?
Not really, because again, the idea is that the network itself represents all the possibilities. So in a way, the network, the network itself represents everything, all the possible scenarios that can happen in all cases. And what we observe in a particular experiment is essentially a subset of the network that is active at a particular time point.
Nothing is created or destroyed. It's just a connection.
So, yeah, so connections are not, I wouldn't say they are destroyed, they are still possible, no, because the idea of the network is all the possibilities to cover all the feasible interactions. That's usually what interaction databases contain. But then we want to kind of, observe at a given time point what's happening.
I would really appreciate to see if you can send some about the article about the COVID. Very interesting. The great work about the COVID, the last part. If you could send the article, please, the one that you mentioned.
I'm sorry. Okay, okay. So, we are running out of time, but I have a very short question about the first part of your talk. I think that's very interesting to focus on the signaling network, and I would like to know if have you think about using this strategy to study cancer, that is a disease very related to regulation, to dysregulations.
Yeah, of course. Yeah, of course. We would very much like now to extend this kind of strategies analysis to more data, including cancer. I would say the most, the main hurdle is usually time and people. So it takes a while to analyze so many different data sets. So we had several years to analyze bone diseases. It would take probably a new person and a new project to be able to do the same with cancer. But yeah, I think it would be a very good, idea. Yeah.
Great. Maybe I will make a contact to you soon about that. Great.
Thank you. Yeah.
Thank you. So thank you very much. It was really a very good discussion. Now I would like to remember to thank you again. So now our next talk is Luciana Barros. Let's see how, okay. Is Stefanella, I think that I need you here, please. I need you, please. Yeah, I don't know how to deal here. I wanna take the stage to Luciana, but I need you.
No, it's here.
Okay, great.
1 second. Can you listen to me?
Yes.
Okay.
Luciana.
Não aparece aqui. Com que não entrou?
Eu não sei, mas ela, she's already here, but we wanna.
1 second. Yes, aqui. So goes to more.
Yeah, Luciana.
Goes to co-host.
Co-host, okay.
I think I can share my screen also.
Yes, now you can share your screen. Let's take here my my comments about you. Okay, great. Now you'd like to introduce Dr. Luciana Barros. She graduated in biological science from Universidade Federal Fluminense. She holds a master and doctorate degrees in cellular and molecular biology from Institute Oswaldo Cruz. She works with immunology, molecular biology, and bioinformatics with an emphasis on cellular immunology. She also works in many topics like T lymphocytes, muscular dystrophy, integrins, microRNA, viral infections, thymic epithelial cells, analysis of new generation sequencing data, genome editing using non-viral vector such as a transposon, CRISPR, and integrases. She currently works with bioinformatics in the area of oncology, genetics, and tumor immunology. So a lot of things, Luciana. So thank you for being here, and you have the stage.
Thank you. Thank you for the invitation. Well, I will change dramatically the subject because, as you can see, I am a biologist and immunologist. So I will change to cancer therapy. So, well, my title I just changed to English, so it's CAR T-cell therapy from medicine to model. I'm recently in São Paulo. I'm a researcher at ICESP, that is Instituto do Câncer do Estado de São Paulo, an institution part of USP. Well, okay, so for you that are not familiar with CAR T-cell immunotherapy, in the last 20 years, immunotherapy has changed dramatically the way we treat cancer. So cancer patients have improved life quality, and we can now cure patients with metastatic glioblastoma or leukemia. So it was approved in 2017, this new immunotherapy. There is chimeric antigen receptor T-cell that you don't need to understand, but we call them CAR T. So what is CAR? The CAR is a receptor that we made from an antibody that recognizes a tumor cell. So we make like a Frankenstein, we put an antibody molecule in a T cell. So we made this chimeric cell. And then we treat the patient with these cells. So this new T lymphocyte will kill specifically the tumor cells. And then these cells are from the patient, so they travel all the body and will find the tumor and kill the tumor cells. So, and also we can do this in the mouse model. So I also work with mouse. So we have a mouse model where we put cancer cells from leukemia patients, and also the CAR T cells from humans. So we treat these mice with CAR T cells and try to cure them. That is how we test if these cells are good or not, and then we move on to the patients. So I recently made a systematic review of these clinical trials that are using CAR T cells. So basically we have a hotspot on China. China is doing a lot of trials on this matter, and followed by United States and then Europe. And we are here, we are not here yet, but last week it was approved for Einstein to treat and to start the first clinical trial on Brazil. Also, AC Camargo and USP Ribeirão Preto will already started to do these studies, and Butantan will start to produce these cells. So we are moving fast to retrieve this late start. So what we did, it was just to take the clinical trials website and check what is going on in the literature. So I studied almost 1,000 studies that it's ongoing, and they are very early phase one that we are seeing if it's safe or this therapy is safe, what's happening. They are recruiting. So if you have a leukemia or lymphoma relative, you can enter on the clinicaltrials.gov and see if there is a study suits you, and you can be a candidate for a clinical trial. And then how these CAR T cells work. So we have approval for hematological cancer. Well, it's blood cancer, right? So blood cancer is very homogeneous, and these CAR T cells has one receptor that will find these tumor cells and kill them. On the other hand, we have also solid tumor, as melanoma, as liver cancer, as breast cancer. So the challenge here is that we have a lot of different types of cells that are made, that compose the tumor. So the solid tumor is heterogeneous, and it's very tight connection. So CAR T cells to kill tumor cells is very, very hard. And then we can have tumor escape. And then we start to combine different approaches to try to kill this solid tumor that I will get there at the end of my talk. So we also check the different targets, the different proteins that we use as target for CAR T cells, and the different types of cancer. So we have a lot of types of cancer, and then we have the targets. So mesothelioma, most things are the most common target for solid tumors. And for the blood cancer, we have leukemia and acute lymphoma. So we have the approved targets. There is CD19 and BCMA that is also already approved for this hematological malignancy. So that's why there are so many clinical trials here. So what we do in the lab? Well, we start to inject the tumor on the mice, and then we wait a little bit, a few days, and then we put CAR T cells. And these CAR T cells will search the mice and will kill tumors. And here we can see how mice will survive. So if we not treated the mice, the black line, the mice will die in 45 days. If you will treat with a treatment that is not CAR T, it's just T cells, it's an old treatment, we can improve mice survival, but they will eventually die. But if you put CAR T cells, some mice die, some mice will survive and be cured, will be cancer-free. So it is exactly what happened in clinic. We have cancers that we can cure patients and cancers that we cannot. So me and Regina, that is from LNCC, we started a great idea of creating a mathematical model of CAR T cells treatment. And we made this model with tumor cells, of course. So we have a blood tumor. So this tumor has a logistic growth, and they grow and they have interspecific competition, and also they are killed by CAR T cells only. And we have a second equation. So the CAR T cells will proliferate at a attacks. They will die. It's a very small number of cells that will survive. They can kill tumors, and they are inhibited by tumor cells. This is a very important point because in some patients, the therapy are not working, probably because the tumor is inhibiting our therapy, our cells. And then to complicate, we add a new equation that we call memory CAR T cells. So we now, after COVID era, we know that vaccines will generate memory cells, right, from your immune system. So every virus, every tumor will generate these memory cells. So we put that on the model. So we have the effector cells that will kill the tumor, but a part of these effector cells will become memory cells. So these memory cells do not proliferate. They die at a very low rate, but once they found new tumor cells, they will kill these new tumor cells. So when you have a contact with the virus or tumor cells again, you will kill them very quickly. So we have these three equations of models. So what we did, we get data from literature, and based on these experiments from these graphs that is representing the tumor growth in mice, we get our model. So we have the tumor growing here. So if you do not treat the mice, the tumor will grow and the mice will be killed. If we treated the mice with the not so good treatment, some mice will eventually die. But if we give them CAR T cells, they will eliminate the tumor, they will be cancer-free. And in the graphics here, we have the CAR T cell expansion. So it's a living drug. Once we inject them, they will proliferate in the host. So they will grow and kill the tumor, and then they will shrink and they will form memory cells. So we replicate this with our model, and this is the simulation. So the tumor exponentially grows. And then once we injected CAR T, the CAR T will kill the tumor and they will change to a memory phenotype, that is the blue line. And now we get a model so we can make it several tests. So if you have in the mice like 1 million tumor cells, we can kill them all. But if you have 20 million cells, we are not able to kill. And the tumor will eventually kill the mice. So we made all these combinations, and we can see when we have a complete response, we can cure the mice, or when we have tumor escape. So here the tumor wins, right? And then we can also see if the tumor is rising again. We can see that our cells can kill the tumor again. So this is from another experiment, and we can replicate the data because we have memory cells in our model. So if you have another tumor that is the same tumor, your CAR T cells will kill this tumor. Well, the same model also applies to another type of cancers, of blood cancer. This is very aggressive, so the CAR T cells are able to control tumor growth, as we see here, but actually they inhibit so much our CAR T cells that the tumor will escape. So we can see that using the same model, we have easy to treat tumor or a very hard inhibitory tumor. So we can apply this model. We also built virtual populations based on data. So what we do was to change a little bit the parameters, around 20%, and we checked the endpoint, the outcome, if the mice dies or not. And then we got all the simulations that ranges in biological data. So if the mice in the simulated model, mice died before, we discard it. If the mice lives forever, we discard it. So we get the same interval that we have in the biological data. And so with this virtual population, if you inject different doses of these CAR T cells, we have different outcomes. So if we have the standard dose, we have 95% of confidence that the mice will survive. But as we decrease the dose, the mice will be, the tumor will escape and the mice will die. And also we did a sensitivity analysis to see what are the factors responsible for the outcome. So of course, tumor cell growth rate, if the tumor growth rate is too high, the therapy will not be effective. And also the memory, the conversion of effector to memory is very, very important in the early days of treatments. So this sensitivity analysis, I as a biologist, I like it very much, even though I know it's hard to do. So, and also I asked them I want to run the model, but I don't know how to do a mathematical model or how to program a mathematical model. So what we did was to develop a tool. This is online. So it is available, it's a source, open source at GitHub or Zenodo, but it's also available at LNCC website. So it's cartmath.lncc.br. So you can enter and do some simulations. This is the standard set, or you can change the basic parameters or the actual model parameters. And you just click on the run simulation and the model will run. So you don't need to know how to program or anything. This is for biologists, right? So we have the rapid growth, the Haji cells, so we can replicate exactly the paper figure. You can change a little bit, see what's happening. And then we you can also export custom access, custom graphs. So if you don't know how to do the graphs, you can customize your graph and just export. Or we give you the table so we can build your own graph if you know how to do it. And it's very user-friendly. And also, as we run a lot of simulations, we just forget what parameters we use. So the program has a report that you can download. This is a PDF, and all your parameter set, the graph, and the table will be available for you, so you can reanalyze later. So this is the paper that was published last year in cancers. We are now putting more upgrading CAR-T-math. So also there is a manual. You can see how to use, how to do all this stuff. And well, this is the contact for the TMG, the Tumor Growth Modeling Group at LNCC. And then we move on to patients. So, well, this is the CAR-T-math is a very simple model using preclinical data, that is mouse data. But for patients, they are very complex. So this is a very nice story that a little girl with 5 years old, and she has a very aggressive type of cancer, and her parents know there is experiment on mice that was okay, that was good in, and they asked for the doctors to be treated with CAR T cells in 2005 or 2007, 10 years before the approval. So she was the first patient to be treated with success of CAR T cells. So her name is Emily. She has now an institution, and she is already 10 years cancer free. But and she has ALL. Well, for children with ALL, that is acute lymphoid leukemia, you have 90% of getting cured. Even if you have like a bone marrow transplantation, if you have 4 cycles of chemo before. But if you're an adult and you have leukemia, you have 50% of success. So 50% of patients will be cured, 50% of patients will be not. So what is the lottery? And for other disease that is chronic lymphoid leukemia, that is an old adult disease, the response is not so nice, but they respond. So we have a little bit difference between disease and we have a difference in response. So how this is done in patients? So first, the leukemia patients will be treated for leukemia. Disease cannot be out of control. So we get the blood, and then we put the receptor on the cells of the patients. So it's your own cells. And we expand them in vitro in the lab, and then we kill all cancers, try to kill all the cancers, and put the CAR T cells on the patient. But in this process, there is a few weeks to a few months, like from a month to 6 months in production. And we have a lot of reasons that this product will not reach the patient. So if the patient is too ill, we don't have enough cells to produce these CARs. If the tumor grows before the production, the patient will not be treated. If the patient was treated too many times before, if you have like 5 cycles of chemotherapy, the cells can be poor, so they will not proliferate, they will not expand in vitro or in vivo. So we have a problem of cell survival after we inject in the patients. These CAR T cells can be exhausted. They cannot work properly. And then the tumor can change and escape. So there is a lot of things that happen, so the tumor will escape and eventually the patient will die. So for this patient model, we change a lot of parameters. We maintain our structure of effector, memory, and tumor cells, but we need to add 2 more populations. We need to add a CAR T that is distributed along the body, because in the mice, we don't see a delay of function. But in humans, depending on the disease, we have a delay between the injection and the effect. So we have this new population here. And also we have the exhausted cells. They will not kill the tumor. They are there, but they will not kill the tumor. And then this part of the cells that we infuse in the patient will engraft, become effector, kill the tumor, and become memory cells. And the tumor will try to avoid this effect with immunosuppressive. So how do we know that? Because we measured in vitro. In this model, we have several kinds of phenotypes for CAR T cells. And then we have different sorry guys, I'm just recovering from COVID, this time of pandemic. So we have different patient profiles. So we have patient 1, very high proliferation, very low persistence. We have patient 2, high proliferation, medium persistence, and patient 3 phenotype that is low proliferation and high persistent. Which is better? We don't know, we need to test. This model is ongoing, so I just to illustrate what we are doing. So here is the patient data, the points, and the line is the model. So we can see that with our model, we can recapitulate the tumor and CAR T cell dynamics. And then we separate the CAR T cells populations into the 3 that we are studying. So the red line is the 3 populations. And here we can segregate them in the 3 that we are interested in. So, and at the bench, what I did some years ago was, if we do not expand these cells in vitro, if we just make the cells and inject them in the mice in the same day, so they will not become exhausted. And it really works. It will not cure this kind of tumor because this is very aggressive. But if we compare with non-treated mice, it had 50% of better overall survival. If we compare both, we have a still very nice effect and a better product quality. So back in the figure, that all the results I showed you was blood cancer. But now what happens in the solid tumor scenario? Well, the main studies solid tumor is glioma, there is brain cancer. We have advanced solid tumors, that is because one target can target a lot of tumors. And pancreatic cancer, lung cancer, all very aggressive cancer types. So glioma and pancreatic cancer are hard to do surgery. So we decide to make a model for glioma. So in this model, glioma is here, it's a brain cancer. So it's hard to do surgery, of course, because patients will lose some capacity depending on the brain region. And also the treatment nowadays is just with the radiation. So we irradiate the patient's brain. And there are several clinical trials going on. So they inject the CAR T cells inside the tumor. So they make a small hole in the tumor and they inject the cells inside the tumor. So we don't need to migrate to the tumor, they are already there. So what we do? We have CAR T cells, they proliferate and die, and they will kill tumor cells. However, the brain is not composed exclusively from tumors. So eventually we have the glial cells. The glia is cells that sustain neurons in the brain. So in the brain, we have neurons and glial cells. So, and glial cells have the same proteins as the tumor. That's why it's glioblastoma, because it's a tumor of the glial cells. So we have healthy cells that can be target of CAR-T, of our therapy. And we have cells that will not be target of our therapy. So to treat the tumor, we can eventually kill like half of the patient's brain. That is chemotherapy do, radiotherapy will do. We try to be more precise. And also, if the glial cells died, the neurons that they sustain will also die. Because if they are not fed by the glial cells, they will die. So we have on-target effect, that the desired effect on tumor cells, but also we have off-target effects, that is our drug is killing healthy cells. So this is the equations, and this is already recently published in Frontiers. And I'm sorry, I don't know even the Greek letter names, but I deal with this part. So the actual treatment for glioma, as I told you, is radiotherapy. So patients will receive chemotherapy and then radiotherapy. And our model just simulates this treatment. And here we can see the healthy cells. They die, but then they proliferate. The problem is that glial cells can proliferate. However, neurons cannot. So once the glial cell dies, neurons die and they will not be recovered. And the tumor will also die, but they eventually escape. Well, they always escape. We cannot cure brain cancer still. But if we treat a tumor and with chemotherapy, we have these waves that is the chemotherapy cycles. And what happens with CAR-T? So when we treat tumors with CAR-T, the tumor cells will die, and then they hit CAR-T cells, and then they will slowly grow again. What happens with neurons? Well, they will die, they will stabilize, but they will die, continually dying, because tumor is competing for the same resources. So neuron will be defeated by tumor cells. So in this article, we made a lot of simulations with different doses of tumor and different doses of CAR-T cells. So here I will just show you one tumor dose that is 0.1 capacity support. So we have a very small tumor and a lot of CAR-T cells. This is a lot. So, but if we test different CAR-T cells dose, if we put small doses, if we put small doses of CAR-T cells, the tumor will grow. If we increase CAR-T cell dose, the tumor will shrink. And what happened with CAR-T cells? Well, if we put higher doses, they will proliferate and then they will shrink. Remember that they need tumor cells to proliferate, and also they are inhibited by tumors, so they will start to die. And this is the glial cells, the supporting cells. So if they are targets of CAR-T cells, they will be killed, depending on CAR-T cell doses, of course. But if they are glial cells that are not target, they will proliferate and occupy the niche from these cells. If these cells die, these glial cells will occupy the same place. And then we did a sensitivity analysis. So what is the most impacting factors on all these dynamics? Well, for tumor, it is the tumor growth rate. If it is too high, of course, the tumor will always escape. If CAR T cell proliferation is not enough, the tumor cell will grow. So we need a very proliferative CAR T cells. They need to grow. And if the tumor cells have a very immunosuppressive rate, they will eliminate CAR T cells and they will proliferate. So this is what happened in a patient. They are very immunosuppressive microenvironment, and they inhibit CAR T cell proliferation. So our model is according to the literature. So this is just a very brief resume. So we can model CAR T cells from animal models. We can see by sensitivity analysis what is the most important parameters. And we can make a model as a tool to non-mathematician people, so like me. So I ran CAR T math. And also we can model and use these tools to better understand the cell's properties. So I like to thank LNCC. Emanuele made the blood cancer part. Daniela made the glioblastoma part. Gustavo made help us, Gustavo, Andrea, and Arthur help us with CAR T math. Brandon, it's a best student that just start this work, and I'd like to thank you, Regina, for all her support. Thank you.
Thanks, Luciana. Very good talk. Congratulations. I would like to know if we have questions here, or if we have online questions. So okay, I have a question. Very promising result you show. You try to know how you make these cells to be specific, because they have the ability to kill cells, and we must avoid them to kill healthy cells. How make them to be specific?
Well, we use antibody properties. If you have a very good antibody that recognize a target, we use them. But this is really hard to choose the better target. So for leukemia and lymphoma, that is blood cancer, we kill blood cells. They are not the most important to our survival. You know, if you kill your lymphocytes, we will live. You just have a problem if you get an infection, but you will live. So actually, we kill healthy cells. There is no way to avoid that. But we try to kill not essential cells, which is hard when we talk about brain cancer, pancreatic cancer, that we will affect very important health cells. So, and now people are trying to combine more than one target. So if you have two targets in the same cell, you are almost sure that will kill tumor and will not kill a healthy cell that is just one target. So this is the tricky part, how to kill the tumor and how to maintain your healthy cells alive. So when you have HIV or HPV cancer, related cancer, it's better because your target will be the virus. So virus proteins are very different from ours, but cancer cell proteins are the same from the healthy cells. This is the tricky part.
Okay. So, thank you again, Luciana. Congratulations.
Thank you.
Now we have a half-hour break for the coffee break. Then we will be back at 5 p.m. with Dr. Miguel Rocha. See you later. Como é que eu faço? Dá uma pausa?
Testando.
Aqui, cara, viu? Boa Olá, boa noite.
Boa noite.
Boa noite. Está me ouvindo?
Sim, sim. Estão a ouvir bem também?
Ótimo. Daqui a pouquinho, daqui a alguns minutinhos, nós estamos começando.
Ok, quando quiserem.
Permissão. Exato. Nós já vamos começar, tá? Aí eu vou fazer uma pequena introdução e você pode seguir com a sua apresentação. Muito obrigado mesmo por ter aceito o nosso convite, tá? Por estar aqui conosco hoje. Muito obrigado mesmo. Muito obrigado.
Eu é que agradeço o convite.
Okay. Hello again. It's my pleasure to introduce Professor Miguel Rocha. Miguel Rocha is an associate professor in the informatics department and in the Center of Biological Engineering at the University of Minho, Universidade do Minho. He is also the executive director of the Bioinformatics Laboratory in the Center of Biological Engineering. Professor Rocha, thank you very much for accepting our invitation. Thank you.
Yes, thank you, Fabrício, for the nice presentation. Thank you for inviting me. It was not the best of the days since I am in a conference in Italy, so I hope the hotel Wi-Fi is good enough for the talk. If there are any problems, please let me know. So I'll start sharing my presentation. Please just let me know if everyone is seeing the presentation.
Yes.
Okay, so thank you. So I will try to talk a bit about the work that we have been doing in our lab. So our lab is part of the Center in Biological Engineering where we do our research. I'm a computer scientist from my background, but I've been working in bioinformatics and systems biology topics for the last almost 20 years. And we have been able to have, so it all started as a collaboration of computer science and biological engineering at the time in 2004 when I finished my PhD, and we managed to create a group that has been always above 20 people for the last, I would say, at least 15 years. And the main areas where we work and the main topics that I will discuss a bit today is constraint-based models, so part of the systems biology topic where we do applications in metabolic engineering and else topics, and also probably more recently I've also started to try to combine this modeling with machine learning and data sciences to be able to have data-driven approaches that can complement our work in modeling. So today I will focus more on the modeling part with some help from artificial intelligence optimization engines to help in some problems. And of course, this isn't not done alone. We have long-standing collaborations with many people around the world. These are the ones that are active now. The map would be much more complete if we include all past collaborations, and we have also collaborations in Brazil, mainly in UFSC in metabolomics. So in our group, this is the slide where I try to explain a bit what do we do in our group in a way that we can explain everything together. And basically the idea is that we build this stack of methods that allow us to face different problems in biology and in biomedical applications. And our idea is that if we are able, so this is our grand truth, if we are able to model a system well, we normally can devise good prediction methods, and with those prediction methods, then we have the tools that can allow AI to do their work in terms of optimizing systems. And so we have this stack, so trying to have accurate models of the systems that we are modeling. And of course, since we are working with models, this is more, I would say, more engineering than biology. We have also to define assumptions to the things that we model. But these allow them to make predictions. Of course, the predictions may be right or wrong, may be more accurate or less accurate. Of course, we like them to be as accurate as possible. And when we say biological systems, we mainly are talking about cells. This is what I will discuss mainly today. So models of different types of cells. But in our group, we also work with more data-driven models, for instance, of simpler things like molecules, proteins, compounds, and so on. But in this talk, I will mainly be talking about models of cells that allow to predict phenotypes and that allow them to do cell optimization with different approaches. And then we have the tools. So how do we do this? How do we model? How do we predict? And so basically we have in the side of bioinformatics, systems biology, we have different modeling paradigms. For instance, we work a lot, and I will discuss a lot today, constraint-based modeling approaches, but we also have other approaches like kinetic models and so on. But we have a strong group in reconstructing metabolic models based on constraints, and then also using pathway analysis tools to explore those models. And then in the blue, in the left side, we have our AI, so we have artificial intelligence. And what do we do with artificial intelligence? Mainly we use some tools for data integration and text mining that can help in improving our models. We have machine and deep learning. I will not discuss this today, but we have machine and deep learning that can help also to create models, for instance, of proteins and compounds, but also to improve on our cell models and to have good prediction methods, or to help in having good prediction methods. And one thing that I will discuss today is how we can also use AI to get some tools to do optimization of cells. And in our group, we have worked a lot with evolutionary computation, and I will show you a bit on this later. So with this let's say, this approach, we mainly address 3 types of research questions in our group. The one I will focus more today is how to optimize cells to be able to reach phenotypes of interest. So what can we do to change cells to make them work our way, to make them do things that we like, for instance, in metabolic engineering, but also in some applications in health. But we also have in our group, and if you are curious about that, then send me an email and I can send you some materials on this, on these other 2 questions: how to develop novel pathways to produce some desired compounds that may be novel compounds, and also how to create novel compounds with desired activities. For instance, we are working a lot on designing new sweetener molecules. This is something that we have been doing in the last years, but I will not have the time to go to that part today. So if you have any interest on that, just let me know and I can send you some material. So with this approach, with systems biology combined with AI, we also have different application areas from the side of biomedical applications. I will focus today on 2 main applications, one in metabolic engineering. This is what we have been doing in our group since its beginning, but we also have some applications in cancer metabolism that I will discuss a bit, which are more recent, but they are also, I think, interesting to show these approaches of combining AI with systems biology. Just to let you know that we are also involved in big projects. So this is an important project for us because it's also led by our spin-off company that we created from our group in 2010, and that is now leading this 8 million euro project from the EU. And this project is interesting because it shows all the 3 different questions that I mentioned before. So this project includes all of them, and the idea of the project is to have effective cell factories for the production of 100 different compounds that have as in common the fact that they can be produced from the hub of the shikimate. So the shikimate pathway is a common shared pathway for all these 100 different compounds. And the idea is to show that we can do metabolic engineering, sustain optimization in large scale for all these products, but we also do pathway discovery in this project, and we also are creating novel compounds, in this case the sweeteners that I mentioned before. So it basically addresses all the 3 questions that I mentioned. So it's a kind of a flagship project for our group and for our spin-off company. And it's also interesting because it shows how we normally need to work in this area where we have the in silico computational approaches to design molecules, to do strain optimization, so to design new pathways. So there is a lot of computational work, but there is also a lot of experimental work, work in molecular biology to change the genetic background of the organisms, so to do knockouts, to introduce heterologous pathways and so on. And there is also lots of work in validating then the strains that we create from, I would say, small scale to large scale fermentation labs. So, and we have in the project partners that can do the wet lab implementation of the solutions that we create, but also then to scale up to real fermentations. For instance, DSM is one partner of the project that can then take the strains that we are creating for these 100 products and make them products that can go to the industry and be effectively implemented in industry. Okay, so let now try to show you a bit on what we do in cell optimization, and because this slide pretty much what you already know. So we have omics data, we can do a lot with new high throughput technologies. And one of the things that we can do is to try to create models based on all the data that we have now available, from genomics to other types of omics data that can characterize phenotypes. And there are two things that I will focus today. One of them is that I will mainly discuss modeling in terms of metabolism. So it's a subsystem that we study more because it's the most important for metabolic engineering applications and others in health, but also because it is probably the most well studied subsystem where you have more data and you have more information available. And within metabolism, we will discuss mainly constraint-based approaches. So these are the ones that allow you to work at the genome scale level. So we try to model not small parts of the cell behavior, but we try to model cells as a whole. So we try to model all the metabolism of the cell. So we try to work at the genome scale. So try to work with all the genes that encode enzymes and to all the reactions in an organism. And to do that, constraint-based models are for now still the only available option, because if you try to do kinetic models of genome scale size, it is very difficult to work with these models to have a good parameters for those models, because they require much data to be well tuned. So we still mainly work with these constraint-based models that have, of course, some limitations, and I will try to discuss a couple of them. But they have a big advantage, is that they allow you to work at the genome scale level. Okay, so basically the work that we have been doing in our lab for many years is mainly on strain optimization, and what we try to do is to have a rational approach to strain optimization. And what do I mean by rational? What I mean is that we try to have information from the genome that can be used to model metabolism and have good models of metabolism. And those models of metabolism will be used as the basis to define what are our optimized cells. So the way that we think of in this part is we try to use models to propose alterations in the genome that are rational given the model and the knowledge that we have on the system. So this is not done, for instance, with mutagenesis or in a random way. So we try to propose modifications in the metabolism that make sense based on the knowledge that we have on the system and on our models. Of course, then you go to the lab and some things work, some things don't, but you have a rational basis for that, and you are not doing things in a blind way. Okay? And why does this make sense to do strain optimization in the first place? Normally it's because even if you have, let's say you have an E. coli bacterium and you want that bacterium to produce, let's say, succinate, and you go to the metabolism of the bacterium, and it is really possible to do it. So the bacterium has all the machinery to produce many things, but in the wild type, so without any changes, it probably won't. So if the organism is not in a stressed condition, if it is growing, let's say normally, it will produce only a small fraction of what it is able to produce. So if you want to convince, let's say, a bacterium to produce succinate or another product of interest, you need to change something in its metabolism to make it produce that compound without any stress conditions. And normally the trick that we try to do here is to convince the bacterium to do it by forcing flux redistribution. So we try to cut or reduce flux in some pathways that are normally used for a maximum growth wild type strain, and we try to couple growth with other pathways that may be more interesting to produce byproducts that we might be interested. Let's say we want to produce ethanol or succinate, then we need to convince the bacterium, the yeast, or whatever to use the pathways that do the production of those compounds to maximize growth, and that really needs to retrofit the metabolism to be able to do it. Okay, so the idea here is that what kind of genetic changes can we impose the host organism to be able to produce the compounds of interest. Okay, so that's the basic idea of what we do and the justification for what we do. So the stack is basically what I've shown before. So we have these three parts, try to create good models of our host organisms to be able to do phenotype simulation. And since we are now talking about metabolism, phenotype simulation means normally fluxes for reactions, and then we can use AI, for instance, evolutionary algorithms and other heuristics to optimize. Okay, so basically here what we want is we want to maximize the production of a compound. The wild type strain doesn't do it, so we need to see what kind of genetic conditions can we do. So, for instance, gene knockouts, for instance, to do over under expression of some genes in the host to make it a better producer for our compound. And this framework is very generic. You can see that we don't define here what kind of model we are using. It can be constraint-based, but it can also be a bit more complex, and we will discuss a bit some of these alternatives in the next of the talk. And we can have very different phenotype simulation methods that can have their own assumptions. And one important thing that I want to mention since the beginning is that in modeling, we always have assumptions. So we are always, when you do a model of any system, you always simplify the system. And in biology is not an exception. So when we are modeling biological systems, we simplify. So we create assumptions that allow us to reach conclusions. But those assumptions are assumptions. They are not ground truths. So they are not something that is indisputable. So they are assumptions that sometimes are true, sometimes are not true. And you need to be careful on what are the assumptions that you can assume, and to be careful on the results to know that you have assumed that those assumptions. So in the end, the result will be based on that. So if the assumptions are simply wrong, the results are not to be trusted. Okay, and that's very important in all these modeling approaches. Okay, so a bit on this stack. So we in our group, and of course many people in the community, have been working in all these three levels to improve models, to improve phenotype simulation, and to improve the optimization part. For the models, this is a process that is quite standardized now, but still in many cases it requires some feedback from the lab and some manual curation of many things. But it can be semi-automated in many of the steps. So basically what we do, if we want a model of a new organism, we go to its genome, we annotate the genome, we identify what are the enzymes in the genome, what kind of reactions can be done by those enzymes, the stoichiometry of those reactions, and then we also try to find the reversibility of reactions, compartmentalize the reactions in the different compartments of the cell, and then some more, I would say, a bit more manual tasks, or at least less automated, to think about what are the energy requirements of the cell that are important in metabolism, what parts we want to model or not. And in many cases, it is also important to define what we call a biomass reaction, which is basically what is in the cell, what the cell needs to create, what kind of precursor molecules the cell needs to have, and in what amount to be able to divide. And this is important because it's a way to stimulate cells, is to think that they are trying to grow as much as they can. Okay, from this reaction portfolio, we need, of course, to do some experimental validation. So I will go to this mathematical representation in a bit, but once we have the model and the mathematical representation, we can do predictions, and so we can experimentally validate some of those predictions and try to improve the model based on the results. Okay, so this is an iterative cycle. In our group, we have a very nice tool. It's called Merlin, and it is a tool that allows to create these models and to make it easier for someone that's creating a model to do the manual curation part and try to automate as much as we can all the parts in this reconstruction process. So if you are interested in this area, I would really recommend that you can take a look at this Merlin tool. Version 5 was very recently, a few weeks ago, published in NAR. So it's a tool that's recognized now by the community and with a good user base as well. And with this tool, of course, in our group and others, we have been working on many different reconstructions and models. So these are some of the recent models, although I haven't updated this in a while. So probably there are more recent things. So there are lots of different models being generated in Merlin as we speak. Okay, explaining a bit how we can use those models to make predictions. I will need to go a bit to the mathematics of this modeling approaches. So what I did here is to put a very simple cell. So this is a very simple representation of a possible cell, just to show you how we can model this. Of course, this is not a real cell. And what you see is that you have the boundaries of the system. In this case, we don't have compartments. It's a very simple thing. We just have, let's say, a membrane that isolates our system, our internal part from our external part. And what we do in constraint-based models is to say, okay, all these metabolites that are represented here in the green boxes inside the cell will be assumed to be in a steady state. So what we mean by that is that, for instance, this A metabolite will be created and consumed at the same rate. Okay, so in this case, A enters the cell from the exterior by this R1 transport reaction, and then it can be consumed by R6, by this R5, and by this R10. Okay, so what we can say is that the rate of the reaction R1 that is putting A into the cell needs to be equal to R5 plus R6 plus R7. Okay, so we can, based on the steady state condition for A, define this equation here: R1 minus R5 minus R6 minus R7 equals 0. This R1, R5, R6, R7 are the rates of these reactions. Okay, and based on all these steady state equations for all the metabolites, for B, for C, and so on, we can build this matrix. So this S matrix, S comes from stoichiometry. And basically what we have here is that we put in each line a metabolite. Let's say in this first line it is A, and A has a positive number for the reactions that are producing A, and the negative stoichiometric value for the reactions that are consuming A, and this represents in this first row basically is this A equation down here, and we can do the same for B and so on. So in this matrix we have stoichiometric coefficients for rows are metabolites, and the columns are the reactions in our system. And this framework also allows us to specify if a reaction can occur in both directions, or if a reaction only occurs in a single direction. So we can also define those constraints. And basically what this allows is that, so we have here a mathematical foundation to determine flux values that are feasible in this SV equals 0. So this is what we call the null space. So it is a set of possible fluxes that obey these constraints of the steady state. And we could even try to take a look at this system and solve it to get R1, R2, and so on. But the problem is that you cannot solve this system because you don't have enough equations to get all the possible values. So this is what we normally call an underdetermined system. And so what we normally do is to try to either have measured numbers. So if imagine that you can measure, for instance, this R1, so you know what is the input of this A, or you can measure this R3 and so on. And in this case, if you can measure 3 different, or in this case 4 different values, you would be able to solve this system. So imagine that you can have a value for all these transport reactions that are putting things inside and outside of the cell, so you could solve this system. The problem is that when you go genome scale, normally the numbers of degrees of freedom in these systems are very high. We are talking about hundreds of measurements that we needed to do, and so it is normally impossible to solve these systems for genome scale cases. So what we typically do is transform this into an optimization problem. So what we do is say, okay, let me maximize or minimize something that makes sense. And if I do that, then I will have a flux distribution that will correspond to this objective function, and I can get the flux values. And one of the most used objective functions is simply to say, okay, let me maximize growth. Let's say that I know that my cell has been evolving in over time, over millions of years or over many generations, to grow as fast as it can. So I can assume that it will maximize the biomass precursors. So if I can define an equation that defines this biomass composition, I can maximize this, and I can get the values for fluxes. One good thing about this is that we know that the constraints that we have defined here are linear. So we have defined linear constraints, and this function, the biomass function, is also a linear function of the biomass precursor. So it's a linear objective function, linear constraint, simplex algorithm. We can solve this computationally very easily. So we have a good method, and this method is typically called flux balance analysis, or FBA, and it has been used around the community for a few decades now. But this is not the only way we can think about this system. So we can say, okay, let me think about a different option. So what does happen if instead of maximizing the biomass, I want to, let's say, to simulate a mutant where I did some knockouts of genes. So in this case, it hasn't evolved to optimality. So probably what it will try to do is to be as near as possible the metabolism of the wild type. So we can define that with different objective functions. So there are methods, for instance, this MoMA, this room, that change the objective function. In this case, sometimes the objective function is not linear anymore. So we need to do quadratic programming or MILP and so on. And we can do different methods for this simulation, okay? And until now, I've only discussed reactions and metabolites, okay? But normally when we go to the lab and we do knockouts, when we change something in the cell, we work with genes, okay? So how can we connect these things? And that we can models, something that are normally called gene-protein reaction rules. And these gene-protein reaction rules are basically rules that define how we can reach a reaction based on the genes that encode the enzymes. And for instance, in this case, you see that for these two reactions, you need 4 different genes to be active at the same time. So this is probably a protein complex for this reaction to be active. And we can define these Boolean binary rules for all the genes and the reactions in our models, and so we can have a very simple way to interconnect the reactions and the genes in our system. Okay, and we can even, if we have information about the regulatory network or about the regulation of our system, and now typically FBA doesn't work with the regulatory constraints, but you can have them. For instance, if you know there is a transcription factor that affects a number of metabolic genes, you can connect those with the rules that are very similar to these ones here, but in this case, the right-hand side of the rule are regulatory genes and not metabolic genes. Okay, so we have discussed a bit the first two levels of our stack, the models, and in the middle level, the simulation, the prediction of phenotypes. So now we are ready to discuss how we can do optimization. And the optimization is basically defined in this way here. So the aim is normally to select what are the appropriate genetic modifications that I should do to my host to make it more suitable for our bioengineering problem. Okay, so let's say we want, for instance, to knock out a few genes to make the host to overproduce a given compound. And at this point, I need to explain, because if you are hearing this for the first time, you normally are a bit confused. How can we create better strains by deleting things? Okay, and sometimes we get this confusion, but you need to think about what we want to do is to redirect flux. So we want to convince our cell to use pathways that it normally doesn't in a normal condition. And one way to do this is to cut some of the pathways that are being used in the wild type to force the cell to go through other alternative pathways. It's a bit like when you are watering fields, then to make the water go through certain pathways, you normally close in one side to make the water go through another direction. So this here is a bit the same thing. And so what we normally are trying to do is to maximize a given compound production. So this is our bioengineering objective, the things that people want to do in cell factories. And there is an internal or cellular objective that is the one that we need to use when we are doing phenotype simulation. And here there are two different things. One of them is what do you want your cell to do, and the other is your ability to predict the phenotype of the cell when you do certain modifications. And so this inner part here is what the cell will do, or what we can predict the cell will do given some modifications. And in the outer part, what we are trying to do is specify different alternative genetic modifications so that I can find one appropriate one. Okay, and these genetic modifications can be gene deletions, can be to overexpress or underexpress certain genes in your network, or it can even be to insert heterologous genes from other organisms to make, for instance, you want yeast to produce vanillin, and you need to get some enzymes from vanillin producing plants to make it produce vanillin. Okay, and how do we do this optimization part? So in our group in 2005-06, we have started to propose this evolutionary algorithm approach for strain optimization. And our idea is so evolutionary algorithms come from biological ideas for optimization. So basically what we do is imagine that you have a population of individuals, but in this case, each individual is a mutant strain with some genes missing. Okay, so imagine this first individual is an E. coli where you have removed or have put to zero reaction number 23, 122. So these are indexes, of course, to the lists of reactions in your organism. So here you have three different sets of knockouts. Each one of these is a different strain, so it can be simulated with FBA or another method to get the fluxes. And so what we do in evolutionary algorithms, we define a random population in the beginning. So it has, let's say, 50, 100 in different solutions. Each of these solutions is evaluated. And then we create new solutions by doing something that comes also from nature. So what we do is for instance, let's say we take these first two possible solutions. And in here you see that you have a shared knockout, this 390. And so you can keep the 390 in the new solutions. And then you can select, imagine that you want to create an offspring from these two solutions. You keep the one that is shared, and then you take, let's say, 2 randomly from the first one and 2 or 3 randomly from the second one. So you create a new individual that has part of its knockouts from one parent, let's say, and part from the other. That's what we call doing crossover. Doing mutation is a bit simpler. It's like, for instance, removing one of these values or changing to another one or adding another one, okay? And then you do this process of creating new solutions, evaluating these solutions. And normally what you do in evolutionary algorithms is a bit what it happens in nature. So the best solutions have more hypothesis of being selective, but the worst solutions can still be selected, okay? And for the evaluation part, what you do is quite simple. So you take a solution, you put to 0 all the reactions that are being removed here, and then you add these constraints to your system and solve it. FBA, MOM, or ROOM, whatever, and then you get the flux distribution. So you get the fluxes for all the reactions in your network, and you then apply a certain objective function. Imagine that you want your strain to produce a lot of a product and also to have a higher biomass, so you can multiply those two things and get this BPCY function, okay? And it can be a nonlinear function because here you really can work with any objective function that you want, because evolutionary algorithms are very permissive in all that you can do, okay? And then I'm looking at time because I talk too much, so I'm getting a bit late, so I'll try to make some parts a bit faster. So we have also done this, but instead of considering just one objective, consider 2. For instance, you can, instead of multiplying biomass by the product, you can say, okay, I want strains that grow a lot, or at least grow as near the wild type as we can, and that overproduce a given compound. So I can define 2 different objectives and try to do multi-objective evolutionary algorithms that are around to do that. You can even say, okay, since I have to go to the lab and do knockouts, each knockout requires 2-3 weeks of work in the lab, so I want to minimize the number of knockouts. So I can add a third objective to that. And then I will reach this kind of results. So for instance, in here you have some results with 2 objectives and 3 objectives, and each point that you see here in blue or red are possible trade-offs between the two objectives that you are optimizing. So then someone that's looking at these results can select one trade-off between these 2 objectives and go to the lab and explore that solution. Okay. And okay, with this all this part, we have also done some software. We have OptFlux, which is a nice software for non-programmers, so it has a very nice user interface, and it has been around since 2010, so it's very stable. We are not improving it anymore, so we don't have the resources to do it, but the version that's around is very stable and can be used still with the models that exist without any problems. And in more recent years, we have been working with different things. So we have been, for instance, creating chassis, which are designs in strain optimization that can be used for different products. That's something that we did also in Shiki Factory. And so the idea is that we can do one knockout, and that knockout can then be complemented by a second one to produce, let's say, succinate, or can be complemented by a different one to produce lactate. Okay, so there is a core that we call chassis that can be used for different products in the same pathways. And another thing that we have been also doing is to extend this model. So the constraint-based models are a bit limited in what they can model. So we are trying to account for different constraints and account for the different subsystems in the in the cell. So this includes transcription, translation, regulatory models, and also accounting for enzyme abundance as constraint, because in the cell you cannot have infinite enzymes. So we also try to look at proteomics data, for instance, to try to create constraints that will be related to each enzyme's abundance, or a pool of enzyme abundance, if we cannot get data for all the cases. And in this case, these are, I'm sorry for, there was one thing that was not supposed to be there. One of these approaches is gecko modeling, where we consider our models to have the abundance of enzymes as variables, and so we can then create constraints with this. So we need to extend, of course, the S matrix with enzymes, and to have those in our reactions. We can, if we don't have information for specific enzymes, we can have pools of enzymes, but it is been shown that this approach really prevents some predictions of some of the normal, let's say, constraint-based models. And with all these things, we also have been working in software to support all these modeling approaches. So I mentioned Gecko, but there are other approaches that I'll not mention here, like ME models, S-moment, and so on. And so what we did is to do, since we don't have many resources to do user interface software, we are now doing more Python-based software, where we can create the APIs that people can use in their notebooks and implement all these things. And in this way, we can develop more software and have richer environments where we can have different modeling approaches, and also take advantage on some things like Cobra Pi, framed and reframed, which are already there to make part of the work. So this MuPy package that we have, for the ones that like to program a bit in Python, is very good to try all these different modeling approaches, try different optimization algorithms, different simulation algorithms, and so on. And it also now supports kinetic models. They are still there in the bottom, and it is also implemented already in Cobra Pi. Okay, so here I would finish the part of metabolic engineering, which is the largest one. I don't know if you have any questions on this part.
Yes, I do. We, in our group, we have a project where we collect a large number of bacterial genomes all over Brazil. And sometimes we would like to create metabolic models for some species from very specific cases. My question is, for those tools that we show today, which would be the more adequate, in the sense that I have an annotated genome and I would like to some way to have some support to create a metabolic model? It would be the Merlin.
Yeah. So you have lots of tools to create metabolic models. So Merlin is one of them. The advantage of Merlin is that you can do an automated annotation of your genome, and then you can curate it easily. So you have a user interface that allows you to see why the model has a certain reactions, and if you don't agree with that, you can change easily. You can go through all the, for instance, the BLAST or the diamond annotations that were made. You can even re-annotate, so you don't have to have an annotation. So you can work with the annotation, or what we prefer is to re-annotate all the metabolic part of the system, and then be able to look at it. If you only want a model, a very fast model that is completely annotated, because you are doing that for many organisms, then there are other tools that I haven't mentioned here. For instance, there is CarveMe, which is an implementation of a MILP system, where it's a very fast way to create a working model. Of course, the model has many limitations, but if you need to have a very quick model for hundreds of different things, is the way to go.
Okay, thank you. Thank you so much.
So any more questions on this?
Any more questions from the audience? Not at this time. Thank you.
Okay, so I don't know how much more time I have, so I had here some things on our work in constraint-based models for health. So I'll try to make it short, since I think I only have 15 minutes or so. We have been also working not so much time in constraint-based models for health, and of course the community has been doing that. I would say it's much less stable, or the work has still to show that it can really model well cells from humans. And so it is I would say it's still in its infancy, while the metabolic engineering part is, I would say, much more stable and provides much more results already that you can trust. So it's being used in companies, in industry, and so on. This part of health is still a bit ongoing in research terms, also because it's much more complex to model human cells. Okay, and so the main applications of these constraint-based models in humans have been in cancer, because cancer cells are normally cells that grow, and that where it makes sense to optimize based on FBA, for instance, because cancer cells are normally trying to grow as fast as they can, in a way. And then also to study, for instance, pathogens and those pathogen interactions, because pathogens are typically simpler systems where we can create models the way we saw. And then, of course, metabolic related diseases. And there's been lots of work in inborn errors of metabolism, but also in diabetes, obesity, and also aging and Alzheimer's that have metabolic phenotypes. And one of the things that makes this area promising for the future is that you can use constraint-based models as a way to promote personalized medicine, because you can create individual models for different subtypes of diseases, and even you can create for each individual patient, because if you have different omics data for different patients, you can use constraint-based models as a kind of structure that can be used to overlap a bit your omics data and make them more easier to understand and to get conclusions. Okay, and so since 2007 there have been many metabolic models of human cells. These are general human cell metabolic models, so they keep all the possible metabolism that should be around all human cells, and of course each cell then has its own subset of this. And so the first years of this part were done with these general purpose models alone. So you have here some applications for related to some of the things that I mentioned. So these are early applications of these models. Then people started to realize that it would be interesting to have specific models for specific tissues. It would be interesting to use omics data to influence the phenotype simulation, because in many human cells you cannot really say that they are growing at the maximum speed. So you don't have FBA to work. So you need to have different objective functions to be able to simulate phenotypes. And in that case, normally omics data are used to create phenotype simulation methods that give fluxes that are consistent in a way with omics data. Okay. And of course, here you have several problems, because normally you measure, for instance, mRNA, or you measure proteomics, and then you are predicting fluxes. So there are a number of assumptions that you need to take that are not completely clear. So there is no linearity between gene expression and protein abundance, and there is no linearity between protein abundance and fluxes of reactions. And that makes this much more difficult to work with. Okay, so basically what people did was to try to do one of two things: either to develop new phenotype simulation methods based on omics data, or to use the omics data to reconstruct tissue-specific or cell type-specific models for different, let's say, liver cells, renal cells, and so on and so on, and of course cancer cells. And in this case, of course, there are different approaches. One of them is to see what are the metabolic tasks that each type of cell needs to do and use that in the method. Another one is, of course, to define thresholds over expression data. Another type of approach is to be able to, from literature or previous knowledge, or even expression data, to define a set of core reactions that need to be in a certain model. So there are different flavors of all of this. You can see here some of the methods. Here you have a graph where you can see the methods organized a bit by their different approaches, but this is only to say that there are many methods. Of course, here they are not all, there are many more. There are many methods to do this kind of thing. So this is just an example, and this allowed to focus a bit more on some applications. So we have here some examples: Alzheimer, malaria, obesity, aging, diabetes, and so on. And these are all applications that came a bit later with tissue-specific models already. And of course, cancer is a very common application of these methods, because we are dealing with cells that are growing, with cells that have important metabolic phenotypes as well. Many of the cancer phenotypes are much related to metabolism, although the drugs are not so much. As you know, many of the drugs for cancer are related to signaling, but the phenotypes of the cells show very interesting metabolic phenotypes that we can work with. And so cancer has been a very important application, and also to try to find some drug discovery alternatives. I'm hearing some noise, and someone has the mic open.
Encore lit. Tu n'es pas encore au lit.
Okay, thank you. And okay, so just to give you an idea, so this is the results of a very recent paper that we have on PLOS computational biology, where what we did is to, because we know that there are many difficulties in so this is related to cancer metabolic modeling, but there are many difficulties in trying to really model well the flux distributions or the prediction of flux distributions in cancer cells. And what we try to do is to make it a bit more systematic, to try different methods to reach the tissue-specific models, and to try different prediction methods. So what we did is to develop some software in Python that would make it easier for someone to make these pipelines more, an easier way, and then to try to do some predictions based on some data. Okay, and so this is the first part of what I will show. So we have some interesting packages. If you want someone that's working on this area, we can discuss this and further. This Tropo package that allows to create models based on omics data and a template human model. And what we did was this pipeline of reconstructing, so trying different pre-processing approaches for the omics data, trying different reconstruction algorithms, and then trying to validate those based on different data, and to see how each algorithm and how well we could predict these things. Okay, so I'll go through some of these things. So these are some of the things that we need to solve. For instance, how can we pre-process gene expression data to make it adequate for this work, where we want binary things, either a reaction is there or it isn't, and this is not always easy. How you threshold this data? How do you work with those ends and doors, with isoenzymes, with protein complexes? So these are things that we need to solve. How do you refine a model? So when you get a final model, it needs to be able to. So we normally.
I didn't catch that.
Normally need to.
Sorry, I'm still not sure about that.
Sorry. We need to make the model complete in the way that it can produce all precursors for biomass. So we need to gap fill the model, and we also develop things for that. And then we took CCLE. I don't know if you are familiar with this. So this cancer cell line encyclopedia, which has many different types of data, genomics, proteomics, transcriptomics, and so on. Data on the, hundreds or probably more than 1,000 of different cancer cell lines. And so this is very interesting because we have essentiality data, we have metabolomics data, and we have transcriptomics data to be able to create our model. What we try to do is to create models and validate those based mainly on gene essentiality. So is a gene, in our case, the metabolic genes in our model, are they essential or not? Can we predict that from our model? And then also fluxomics data that we have available for a small part of the cancer cell lines, mainly in breast cancer, and of course also predicted phenotypes with knowledge that is curated from literature. So this is the way that we try to validate. So we created this pipeline based on RNA-seq that would allow then to calculate all these thresholds, all this pre-processing, then gap filling the models to make them work, and then we could try to validate the models based on gene essentiality and fluxomics data that is available from this paper for some of the breast cancer cell lines. Okay, and so this is what we did. So we first used only MCF7 cell line, a breast cancer cell line, and we tried many different models. We tried to see what was the effect of different parameters. So I'll go through quickly on this, and if someone is interested, let me know, and I can give you a link to our paper, and you can take a look at this with more detail. So after doing with MCF7, doing some tests that allow us to see what are the parameters that work best, then we reconstructed models for all the CCLD cell lines, and then we worked with the whole set of the cell lines for the different types of cancer. We took a look first at the ones for breast cancer, and we saw that by doing some PCA analysis, we could identify here the different types of breast cancer, although with some overlap, but the luminal and basal are very well separated. And then we tried also to see if we could use the models as a scaffold to put omics data, and then from the predictions of the models, we could apply machine learning that could work better than machine learning directly applied over the gene expression data. But what we saw is that the predictions that we have for the fluxes are not good enough. For instance, if you use reaction presence, you have good models, good machine learning models. In here, what we are trying to predict is the cell line's primary disease, which is a hard task. So you see here, MCC values are not that big, but the model simulations are not very good features for this machine learning. And so we still are having lots of trouble to predict fluxes of cancer cell lines. And this is something that the community as a whole hasn't been able to solve. So we have, if you see here, I passed that, but if you see here some predictions for gene essentiality, it's already not that bad. So you can predict more or less well if a gene is essential or not. Of course, you still fail in many cases, but in fluxes, you really are not able to predict. So our next step, and I will not mention this because it's even not published, but I will just give you an idea. What we try to do is to extend this model, and we try to, so this is our approach. So instead of going only for the reactions and the genes, we added other layers that allow us to integrate directly transcriptomics data and regulation for all the regulatory and signaling networks that we have for these cancer cell lines. This is a collaboration with University of Heidelberg, with Julio Saez Rodriguez, with a PhD student that finished this year. And so what we are trying to propose is an extended formulation where we put many more things related to everything that goes from transcription to the to the reactions. Okay? And with that, we were really able to improve a lot the machine learning prediction. So with considering all the genes in our model, we were able to get to the levels of the predictions that we had from the gene expression data. Okay? And so I just want to thank, so you see lots of different faces here. This is the work of many people that I mentioned here. And so I want to thank my group and all the funding that we got from different places. So happy to answer any questions if you have them.
Professor Rocha, thank you very much for your talk. Great talk, excellent talk. I will ask the audience questions. I do have a question. We know that the cancer tumor microenvironment is highly heterogeneous. My question is, when you talk about integration with transcriptomic data, you are considering only cell lines, you are also considering data from tumors, and if it is the case, you are also considering single-cell RNA-seq or just RNA-seq?
Yeah, that's an excellent question. In this work, this is only cell lines. So this is a CCLE data sets that only have information on cell lines, and they do all this work on having a huge number of omics data over cell lines, and they are now progressing to organoids with these cell lines in Sanger Institute and all the people that are related to this CCLE project. Which is connected to the DEP map. If you want to take a look, DEP map project from Sanger and some centers in the US as well. But there is some lines of work related to what you are mentioning. I have, for instance, a PhD student that is finishing her thesis now, and she was creating metabolic models of different cells in the immune system and tumor cells from patients, and we are now analyzing some data. We have already also did some things with stem cells and cancer stem cells in a recent paper that you can also take a look in my profile. And there is some work in trying to model the microenvironment, also trying to model, for instance, all the microbes that are in together with tumors, which seem to be very important, for instance, in urinary cancers, bladder cancer, and so on. And so there are lines of work on all of this. There are people working with single cell RNA data. We are working in that project with the immune system. There are people we have an ongoing project that's still starting to try to model the interaction of everything that is in the tumor microenvironment, including the tumor cells, the immune system, and some bacteria. But that's still something that is in its childhood, I would say, because as you saw, and we like to go on very steady basis. So engineering very steady basis that took many years to achieve. And I think in cancer we there, not only our group but the community as a whole, we still need much more to be able to have good predictors of cancer metabolism, even in cell lines. But still there are many people going in different directions in the community indeed. Yeah.
Okay, great. Thank you. Any more questions? I would like to thank Professor Rocha for the excellent presentation. Thank you so much. Thank you for your time.
Thank you.
Thank you. Thank you very much.
Thank you. Sorry for not accepting the invitation to be there in person. I hope you can invite me in another year.
Next time for sure. Next time for sure. Thank you so much.
Thanks a lot for the invitation.
Thank you.